Good morning, everyone, and welcome to AMALearning and welcome to AMALathon 1 2025. Thank you for being patient with us. Apologies, we had some technical difficulties. It's one of the first times we've done a YouTube broadcast, so we did make a couple of mistakes along the way, but we know exactly what we're doing now, and we're going to make a start. So the aim of AMALathon 1 2025 is to complete all of Biology 1 content, Chemistry 1 content, and Physics Paper 1 content. So let's make a start. But just before we do, I just want to say a big thank you, because over the last couple of days we've gone from 400 and something subscribers to 8,500. So I really appreciate everyone that's subscribed, everyone that's joined our WhatsApp channel as well. Really appreciate it. Now, without any delay, let's get started. Because we started 10 minutes late, we are going to finish 10 minutes late as well. We do want to give that time back to you. So, welcome to AMALearning, where you can AMAL your way to success. And what's really important initially before we start is just to explain to you what AMAL actually means.
Providing you with the equation for success. Activate plus memorize plus apply equals learning. AMALearning, you're about to.
Brilliant. So, activate. So when we initially made AMAL and we've started to make we're a small company at the moment. We've currently got an app on the way as well. But AMAL itself stands for Activate, Memorize, and Apply. And the reason we called it AMAL is because when we spoke to all the teachers that are involved in this project, what we found in common was the students that do the best are the ones that do these three processes. So by activate we mean understand everything. So our main aim of today's session and for the next 6 days is to activate, is to understand everything. It's by the end of 6 days, hopefully, everyone will have completed the activate phase. Apologies, just share my screen.
Providing you with the equation for success. Activate plus memorize plus apply equals learning. AMALearning, you're about to.
Yeah, so this particular, the AMALathon is all about activating the brain. It is helping you to understand everything. Now, the way our PowerPoints work is that they are based on specification points. So it starts on specification point 1.1, works its way through every single topic. And that's what our aim is today. That we'll work through one topic at a time, and then what that'll help you do is it'll help you to understand exactly where your gaps in your learning are. So we've had students in the past that used the AMAL method, and this is part of why we used the AMAL method, is that I'll give you an example of my own students. So we had a student just a few years ago. She came to the country when she was in year 9, couldn't speak much English, but we went through that whole process with her, and by the end of it, she actually got a grade 8-9. Sorry, she got a grade 8-7. So by the time she got to year 11, she got an 8-7. And I've had lots of students that have gone through that same process. And wherever a student has an issue, what we do is we go back to AMAL. We go back, have they activated, as in have they understood everything? Have they memorized the key content? Have they applied that learning to a past paper or to past paper practice? All three phases are really important. Now, what we're doing at the moment is we're activating. So our PowerPoints will help with that. And after this whole session is over, AMALathon finishes, what we'll be doing after that is we'll do an AMALathon 2, where we'll cover all of the paper 2 content, and that'll be in the May holidays. And prior to every single exam, we'll have a 3-hour session where we'll be applying that knowledge. So we'll be based on our own analysis. So we've done analysis of all the exam papers since 2018. So we've got an understanding of what kind of topics come up over and over again. And we'll be going through those topics using past paper questions. So what I'll be doing in those sessions, the pre-exam workshops, is I'll show you a question, I'll give you a chance to answer the question and we'll go through the actual mark scheme. Now, today's notes are free, so you can see what they look like and decide if they're right for you. And right now, for those that are attending the AMALathon live, you'll be able to get all of the paper one notes, all the remaining paper one notes for a reasonable price. So if you look at our posts below, there's a post that goes to our WhatsApp channel. All the additional information is on the WhatsApp channel. So if you can follow that link and join the channel, you'll get all the remaining info there. Now, what are the aim for today is, we're going to start with cell biology. So we're going to start from 1.1 and work our way up to 2.3. In chemistry, we're going to finish off atomic structure, but I think we should be able to get through periodic table as well. And in physics, we're going to start with energy. Again, start from 1.1 and work up to around 2.1. Then we'll finish off that topic tomorrow. So how will AMALathon help you to learn? Now, the following is what I would say is the best approach. Number one, I'm going to read through everything on the slide. Then I'm going to explain it in my own words, particularly keywords, phrases that I know are commonly seen in exam questions. Now, where there's clear links to past papers, I'll emphasize that. And what I'll try to do as I'm explaining is I ask questions. Now, if I do ask a question, I'll leave a little bit of think time. I'm not going to be able to leave enough think time for you to write an answer into the comments, because the whole aim is to get through all the content. It's more so you can think of an answer. Now, if you're constantly engaging with the questions I'm asking, it'll help you to stay focused on the session as well. So for the time being, at least think about what the answer is. That'll help you keep your brain active and keep you focused. Now, afterwards, if you find, and now all our presentations, like I just said, are linked to specification points. So when I'm going through the PowerPoint slides, as you're about to see, if you see, for example, 1.1.1 doesn't make sense, or 3.2.1 doesn't make sense, or 5.3.1 doesn't make sense, then you can make a note of that. So you know exactly where your area of weakness is. That means if you don't understand something, you know where your area of concern is, and you can straight away ask a question. If I can answer it during the session, I'll answer it during the session. Otherwise, as you can see in the timetable, we have a question and answer session every day from 9:45 till 10. And that's where we'll just compile all the questions you're asking, and then I can go through them with you. You can watch the video again, because everything we're doing over the 18 hours will be recorded, and you can come back and watch it. And at that point, you can pause it for each question and really take a bit of time answering it. Then we've got our AMAL animations. We've only covered biology at the moment, but we haven't covered chemistry and physics. But for those who are year 10 and joined us, they'll be done by the time you get to GCSE. It means you can go through it in a revision guide, you can ask a teacher, but the main thing is you know exactly where the area of weakness is, and that's helping you fulfill that at the initial phase. The activate phase is helping you understand everything. Okay, so let's make a start. So this is why when we say amaling your way to success, we've tried to turn the word amal into a verb in this respect. That amaling means activate your brain, memorize, apply. So let's amal all the learning now. So we'll start with eukaryotes and prokaryotes. A eukaryotic cell is a type of cell that has a nucleus enclosed within a membrane and contains other membrane-bound organelles such as mitochondria. For example, animal cells, plant cells, fungal cells, and protist cells. Now, the key learning here is the eukaryotic cell is a type of cell that has a nucleus enclosed within a membrane. So it means this nucleus here has a nuclear membrane around it. And also it has membrane-bound organelles such as mitochondria and chloroplasts. So a key question for this slide would be, what is a eukaryotic cell? And it is, of course, a cell that has a nucleus enclosed within a membrane. So it has a nucleus, so all the DNA is enclosed within a membrane, and also it has membrane-bound organelles such as mitochondria and chloroplasts. Now, a very common question that you'll get is a comparative between a eukaryotic cell and a prokaryotic cell. So it's important to know what a prokaryotic cell is as well. A prokaryotic cell is a simple, single-celled organism that does not have a nucleus or other membrane-bound organelles. Instead, the genetic material, or the DNA, is found in the cytoplasm. For example, bacterial cells, archaea. Now, the main thing to understand here is that prokaryotic cells do not have a nucleus or other membrane-bound organelles. Instead, the genetic material, the DNA, is found in the cytoplasm. So you need to be able to compare the eukaryotic cell to the prokaryotic cell. So, quick question, what is the difference between a eukaryotic cell and a prokaryotic cell? So like I said, I'm going to give you around 5 seconds think time. So in your mind, you can at least think of what the answer is and then compare it to what I say. So the eukaryotic cell has membrane-bound organelles, and the DNA is enclosed within a nucleus. But a prokaryotic cell does not have a nucleus, and it does not have membrane-bound organelles. Instead, the genetic material is found in the cytoplasm. So size and structure comparison. So we've done most of this within the previous 2 slides. Prokaryotic cells are much smaller in size compared to eukaryotic cells. They lack a true nucleus and membrane-bound organelles. So they lack a nucleus and they lack membrane-bound organelles. Instead, the genetic material is present in a single circular DNA loop which floats freely in the cytoplasm. Additionally, some prokaryotic cells contain extra small rings of DNA called plasmids, which help in gene transfer and antibiotic resistance. Now, this is something that will be covered in a lot more detail on the topic of genetic engineering. So the main thing here is prokaryotic cells are much smaller. So they're much smaller in size. They don't have a nucleus. They don't have membrane-bound organelles. Their genetic material floats around in the cytoplasm, and either they've got rings of DNA or they've got small rings of DNA which are called plasmids, which help in gene transfer and antibiotic resistance. So here we've got a bacterial cell. It's important to know what each part of the cell does. So let's quickly go through the function. So what is the function of the slime capsule? It protects bacteria from environmental threats. So in a sense, it works just like a cell wall. It acts as a protective layer. The flagella, similar to a sperm cell and the sperm cell's tail, still different, but similar. It rotates to bring about, now it says here bring about environment, but we can cross out, it just rotates to help it move. So if you have already downloaded the notes, you can cross that out. That's a small error, but it rotates to help it move. The plasmid is a small circular piece of DNA in bacteria that carries extra genes. And the genetic material, so here's the genetic material just there, is DNA or RNA, which contains the instructions for growth. So alongside the bacterial cells, you need to know what the structures of the animal and plant cells, and you need to be able to compare all 3 of them. You need to be able to compare bacterial cells to animal and plant cells, animal cells to bacterial and plant cells, and you need to be able to compare plant cells to animal and bacterial cells. So all 3 need to be able to be compared to each other, similarities and differences. So the nucleus controls the cell. The cell membrane controls what enters and leaves the cell. The cytoplasm is where chemical reactions occur, particularly respiration, which is a topic within itself that we'll cover in paper 2. and ribosomes, where protein synthesis occurs, which again is a topic within itself, and mitochondria, which is a site of respiration. And the mitochondria are within the cytoplasm. The ribosomes are within the cytoplasm. Hence, cytoplasm is a site of chemical reactions. Now, in the animal cell, in the plant cell, you've got similar functions for common organelles. So ribosome has a common function. The nucleus has a common function. The mitochondria has a common function. The cytoplasm has a common function. But we also, and the cell membrane has a common function. But we now have additionally a cell wall, which strengthens the cell, adds a layer of protection. Particularly if too much water enters the vacuole, the additional pressure is balanced out by the strength of the cell wall. And the cell wall is made up of glucose. But the glucose molecules attach to each other in such a way that it adds strength to the overall structure. Then the chloroplast is a site of photosynthesis, and the vacuole contains cell sap. So it contains water and other vital nutrients that the plant needs to grow. So let's have a quick look at this exam style question. Now during the presentation and during our sessions, I'm not going to spend too much time on exam style questions, because the exam style questions we'll be doing during the pre-exam workshops. Like I said before, the pre-exam workshops are there'll be 3-hour sessions that I'll be doing before every single exam. So for example, biology paper 1 is on the 13th, which is a Tuesday. On the Saturday, which is the 10th, I'll be doing a 3-hour session covering lots and lots of biology past paper practice. And it'll be biology past paper practice that's linked to prior exams that, based on analysis, the AMAL team think will be coming up in the exam. So in a sense, it's a 3-hour long predicted paper. So for that reason, I won't spend too much time on these exam style questions, but I will quickly go through some questions and answers. So the cheek cell and the leaf cell. So the two cells have a number of parts in common. So on the cheek cell, label 3 of these parts, which both cells have. Now the answer is on the next slide. Because the second part of this question is in the table, write the names of 3 parts you have labelled above and describe the main function of each part. So what are the 3 things that are found in both a cheek cell? Now the key part of this question is when we say cheek cell, what are we actually talking about? We are referring to an animal cell. Now I should warn you all, my writing and my drawings are nothing special. I am in no way an artist, but I'll do my best. And a leaf cell is a plant cell. So what we're doing here is we're writing down what is common between an animal cell and a plant cell. And as everyone knows, what's common between the two is the nucleus, cytoplasm, cell membrane, mitochondria, and it is reasonable to add ribosomes as well. And as we've just mentioned a couple of slides before, the nucleus controls the cell, the cytoplasm is a site of chemical reactions, the cell membrane is what controls what enters and leaves the cell, and mitochondria provides energy for the cell. Now, like I said to you before, we've got 3 steps in understanding everything. One is activate. So we've gone through the PowerPoint slides, this presentation slides. We're now doing our best to understand everything. Memorization. We've now understood what to memorize from this question. We need to understand the cell parts and the functions, and we've applied our learning to a past paper practice question as well. Now, automatically, when you're doing past paper practice, we're also repeating everything over and over again. For example, we've just repeated the function of a nucleus, cytoplasm, cell membrane, mitochondria. We spoke about it as part of the animal cell, we spoke about it as part of the plant cell, and now we've answered a question. It means we've covered it 3 times. So automatically you've now started to memorize it. Your brain has now attached an importance to it. The more you do something, the more your brain attaches an importance to it. It's a little bit like, for example, if I say to one of my nephews who loves to play FIFA, if I ask him all the statistics about one of the football players, for example, Haaland, he'll be able to tell me all the stats from Haaland based on last year's season, the season before, this season, an inform card, and whatever the other terminologies are, because he looks at those stats so often. His brain's made so many neural connectivities in his brain that it has now become something important to him. And because he's gone over it over and over again, the brain has taken it as important and he's automatically memorized it. And that's what we're trying to do here. The more you're going through your content, the more you're memorizing, so the more past paper practice you're doing, automatically you start leaning towards memorization. And your brain takes from short term to long term because you've gone through it so many times. The brain thinks this is important, I need to know this. So another quick question. Bacterial cell is 1 micrometer, the liver cell is 30 micrometers, and the mesophyll cell is 0.05 millimeters. So now we've got a bacterial cell, we've got liver cell, which is an animal cell, and we've got a mesophyll cell, which is a plant cell. So we've got a prokaryotic cell, we've got a eukaryotic cell, and we've got another eukaryotic cell. The next thing we can look at is this, 0.05 millimeters. How do we convert that to micrometers? Because the remaining part of the question is in micrometers. So we convert milli to micro. Now, what you've got to remember is, when you go from a bigger unit to a smaller unit, you always multiply. So we're going to multiply this by 1,000. And that gives us 50 micrometers. Now that automatically makes this question a lot easier. A lot of people miss that it says millimeters and they don't convert anything. And you'll see why that's important as we go through the question, because it says here, initially identify 2 similarities between prokaryotic. So look what we're doing here, similarities. So we're looking at similarities between prokaryotic and eukaryotic. So that's the key word there, similarities. And the similarities, as we know, are they both have a cell membrane. They both contain ribosomes. Now the diagram below shows, again, it's the same 3 types of cells. And we've already established that this equals 50 micrometers. Identify 3 differences between the prokaryotic and the eukaryotic cells based on the diagram above. So number 1, the prokaryotic is smaller. Now, we would not have known that it's smaller had we not, now we would not have known it was smaller had we not converted the millimeters to micrometers. Because otherwise we would have assumed that the mesophyll cell is the smallest. The prokaryotic cell has no nucleus and it doesn't have mitochondria as well. Okay, moving on now. Animal and plant cells. Now this is, again, a common question. Quickly answer a couple of questions that have just come on the chat. So there was one question, what grades is this aimed towards? So I'm covering combined science, foundation and higher, and I'm also covering triple award science as well. So I'm covering everywhere from a grade 4 to a grade 9. In that I am covering absolutely the whole specification. And we are covering, this particular PowerPoint is specific to AQA. So there's another question that said, are the spec points all for AQA? Yes, every single one of these spec points is linked to AQA. So if you go to the, for example, the triple award specifications, it tells you which specification points are for combined and which ones are for triple. So if you look at specifically for this one, biology triple award specification, then you'll start to see that this is linked in with that. So yes, it covers all grades. We'll be covering from grade 4 to grade 9 because we're covering everything. It says, will there be an AMALathon for every paper before the GCSE start? Yes. So initially, there'll be an AMALathon for biology, chemistry, physics paper 1. In May, we'll be doing biology paper 2, chemistry paper 2, physics paper 2. So that's AMALathon part 2. And like I said, as part of AMALathon part 2, we'll be doing the pre-exam workshops, and that'll be a 3-hour session prior to every single exam. So this is a really common question. How do you prepare a microscope slide to observe cells? So you place a drop of water onto the microscope slide. You lay a thin piece of tissue on the slide, and here it's really important to emphasize the word tissue, because what is tissue? It is a group of cells working together. So usually we take a tissue from an onion. We cut the onion in half and take a thin layer of tissue, put it on top of our microscope slide. Add a few layers of iodine, a few drops of iodine solution to stain the solution. You have the objective lens, the lowest one. You lower it down as much as you can. Then you go to the coarse focus and you start raising it and increasing the distance between the objective lens and this part here, the stage. And then you switch to a higher magnification objective lens for a closer look. Now, initially you would use the lowest magnification, view the image, draw the image, and then you go to a higher magnification and again do the same process. Now, you can also use the fine focus knob to sharpen the image for a clearer observation. Now, this slide and the previous slide on how to make a microscope slide, they are questions that are asked commonly but poorly answered, because most students have done the experiment and what they end up writing is what they remember about what they did. They consider it to be a really simple, straightforward topic, and because of that, they answer the question without the key language. So they don't talk about the magnification objective lens. They don't talk about the coarse focus knob or the fine focus knob. So what potential risks are involved in this experiment? How can they be reduced? So it's a common question that's linked to the core practical. So for example, glass slides and cover slips, they can break and they can cause cuts. So it's important to handle it with care. And iodine solution can cause irritation on the skin or in the eye. If it's spilled, it can stain your clothes. So it's important to either wear gloves or use a dropper carefully. And if it does come into contact with your skin, just to wash it off as quickly as possible. So, cell specialization. What are sperm cells and what are their adaptations for their function? So again, some parts of topic 1, as individual topics, they usually come in the foundation exam. However, for higher, they're the fundamental parts of the learning linked to, as in you need to have understood everything from topic 1 to help you answer questions from other topics. So it's important that everyone has that understanding of how the functions, the adaptations of the sperm cell actually are. So it contains acrosomes within the head. So just this part here. And you can see through the arrow just here. So you have acrosomes in the head that break down substances in the egg jelly coat. So the egg cell has a tough outer layer, outer membrane. So these acrosomes, these enzymes, help to break away at it, to cut it down. Then it has a tail, which is for movement. And it has a haploid nucleus, which contains a half set of DNA. So that means when the sperm cell reaches, what that means is when the sperm cell reaches the egg cell, then half the DNA is in the sperm cell and half of the DNA is within the egg cell. And that can combine to make what we initially call a stem cell, which we'll cover in more detail when we get to 1.2. So the main function is to transport genetic material from the male to the female, and which can then fertilize, to transport the genetic material from the male to the female and fertilize the egg cell. So cells are uniquely adapted to carry out specific functions efficiently within an organism. So here we've got our muscle cell. It contains special proteins that slide over each other, enabling muscle fibers to contract. They have a high number of mitochondria to provide the energy needed for contraction. So this is something that's really important, and this is something that we've already covered. And it's a very common question where in exam papers it very simply says, why do muscle cells have lots of mitochondria? And the answer simply is because they're contracting and relaxing, they need lots of energy. And with mitochondria, what process happens inside the mitochondria? So we're looking at this now from a higher level. What happens inside the mitochondria? Respiration. And then that can automatically then link to, so I'm not going to write respiration correctly there, and that automatically links to the process of respiration, the fact that it's glucose plus oxygen, making glucose and oxygen making carbon dioxide and water and releasing energy. Now, the muscle cells are a store for glycogen, which can be broken down to release energy when required. Now, this links to how our body maintains. So this links to homeostasis and links to how the body maintains the right amount of glucose within our blood. So if we have too much glucose, that glucose gets converted by the liver and by the muscles. So the muscles take in that glucose, and the liver takes in the liver cells taking glucose, and they convert to glycogen. Now, just a quick question. Why would glycogen therefore be important for muscle cells? They would be important for muscle cells because when we're running, our muscle cells will use up the glucose within respiration, but eventually the glucose will start to run out, and our muscle cells need lots and lots of glucose because they need lots of energy. So what the muscle will start to do is turn this glycogen into glucose, and so it'll turn the glycogen into glucose, and the glucose will then be used in respiration. So the cells are uniquely adapted to carry out specific functions within an organism. So for example, the nerve cell, it has a myelin sheath which provides insulation to speed up nerve signal transmission. It has lots of dendrites to connect to other cells, and has a long axon which facilitates the efficient transport of nerve impulses. Now again, when we start looking at how nerve cells send information around our body, the reflex arc and our nervous system, we'll be looking at this nerve cell in a lot more detail. So next cell we're looking at is the root hair cell. It has a large surface area to maximize water and mineral absorption. When we say large surface area, what we are referring to is this part here. Because if you imagine if a root hair cell just looked like this, then the part that actually absorbs water is just this section here. But by having the root hair attached, it means all this extra additional surface is available to absorb the water. It contains numerous mitochondria, so to generate energy for active transport. Now we are going to cover active transport in more detail, but what process happens in mitochondria? respiration, where glucose is used. So glucose and oxygen releases carbon dioxide, water, and energy. And the energy then is used for active transport. So already you can see how important it is to understand mitochondria, and it's linked to respiration, it's linked to providing organisms with energy. And this is where a lot of the higher level questions come from. So where in foundation you might just be asked to define or label, from a higher perspective you might be asked to link this to respiration. Now it has a thin cell wall, and again this is something that needs to be understood from a higher perspective. It's got a thin cell wall to allow easier movement of water into the cells. If you've got a thin cell wall, you've got a shorter diffusion pathway, which again will be covered in later slides. Now, next two slides cover the xylem cell and the phloem cell. We will cover these in a lot more detail because in exams it's important to know the difference between the two. It's important to be able to compare the two. Now it has it's a hollow tube, and that facilitates the easy movement of water. Now, when we go through transpiration, I'll explain this in a lot more detail, but I'm just going to explain it briefly now. If that's your xylem, actually let's explain this from a perspective of straw. So if this was a straw and you've got water at the bottom here, when a person sucks on the straw, what they're actually initially moving from the straw is the air particles. Now once we've removed those air particles, as in we've sucked the air particles out, what we've caused in that area is a region of low pressure. Now where the water is, that's high pressure because you've got lots and lots of particles. The second you suck all the air out, you've now got a much bigger difference in pressure. You've got high pressure at the bottom and you've got a low pressure towards the top. So the particles then move into that empty space. And transpiration happens in a similar way. Because you've got leaves at the top of a tree, it's probably better if I did this in green. The water molecules diffuse out through the stomata, and that always leaves an empty region. So that causes differences in pressure. So in a sense, the water osmosis, it moves from a region of high concentration to a region of low concentration. Or you could look at it from a pressure perspective and say it moves from a high pressure region to a low pressure region. And that causes that movement of water to go from the roots all the way through the stem to the leaves and then out of the leaves. And that's why it's important that the xylem has that empty space in the middle. It's strengthened with spirals of lignin. Again, because you're dealing with all these differences in pressure, if you have automatically the low pressure region that gets caused in the middle would cause the outer part of the stem to, in a sense, collapse. So the strong lignin keeps it open. And it provides support and it withstands that water pressure. And also as the water is going up, you would expect some water pressure as well, as in pushing outwards. It has no cytoplasm or organelles to ensure an uninterrupted flow of water. The xylem cell is a dead cell. But like I said, when we go through this in more detail in transpiration, then we'll go through all the differences between a xylem cell and a phloem cell. Now, a phloem cell has a reduced internal structure to create more space for transporting sugars. So with xylem cells, the water only moves upwards. With a phloem cell, it can move in any direction, and that's because it's transporting sugars. And what do we need sugars for? Respiration. So let me change my pen. That is not the best pen to be using. So for respiration. Now, and for that reason then, water can't just go upwards, it needs to go in every single direction. So the sugars which are produced, the glucose that's produced due to photosynthesis in the leaves, the phloem then takes that glucose and it transports it to all the cells around the plant, around that organism. It contains sieve plates that allow water carrying dissolved sugars to flow smoothly. Sieve plates also help with the change in direction. Because what happens with sieve plates, so I'll just give you an example. Actually, let's use the example here. If, for example, I put lots of minerals on this side of the sieve plate, that means I've got less water here. So this would be low water concentration. That means I've got more water on the other side of the sieve plate. So that's high water concentration. So that means the water go from high to low. It would cause it to move that way. So the sieve plates also help with the direction that the water flows in. It's supported by companion cells, which provide energy. So we can automatically assume now, what is the companion cell full of? Mitochondria, which provide energy and essential nutrients to keep them functioning. Now, when you download the notes, it will have the exam style questions there. So I am gonna just quickly go through. I'm gonna skip past all the exam questions and allow you to have a go at that in your own time. Cell differentiation. So again, this will come up in a lot more detail when we cover the topic of stem cells. And it is a key part of the stem cell topic. But we'll do our best to understand it now. And then I'll recap it and go through it again when it comes up afterwards. So as an organism grows, its cells undergo differentiation to develop specialized functions. In most animals, cell differentiation occurs at an early stage of development. Many plant cells can continue to differentiate throughout their lifespan. And when we cover this when we go through the topic of stem cells, we'll be covering embryonic stem cells, adult stem cells, and meristems, the plant stem cells. So we will go through the differences. In mature animals, cell division is primarily used for growth, tissue repair, and replacing damaged cells. Now, one thing I'm just going to very quickly explain here from a human perspective. When we say stem cell, what for human beings or for most organisms, when the egg cell and the sperm cell initially combine, what they produce, so we've got half the DNA, half the chromosomes within the sperm cell, half within the egg cell. When they combine, that initial cell is what we call a stem cell. That initial cell has no specific job other than to multiply and divide. Now, when it multiplies and divides, the process it multiplies and divides through is mitosis. So we get identical stem cells. Now, usually when it gets to the 8 cell stage, what those cells will start to do is specialize. So at this point, they have no function other than to keep multiplying. At that point, they differentiate and they specialize, so they have a specific role. So for example, here you can see that stem cell in the middle has become red blood cells, fat cells, cardiac cells, neurons, epithelial cells, and so on. So it's changed into specific cells. At that point within human beings, it doesn't specialize again. But like I said, we'll go through that in more detail after. Now, a specialized cell is a type of cell that has developed unique structures and features to efficiently carry out a specific function within an organism. These adaptations enable the cell to perform its role more effectively than a general cell. For example, nerve cell, the root hair cell, muscle cell, and red blood cell. So this is what I've just explained in the previous slide. Initially, the stem cell has no specific role or job, but after that it starts to specialize and take on a specific role. For example, for those examples on the slide now. And the same thing happens in meristem cells as well. So, microscopic technology. A common question is a comparison between electronic an electron microscope and a light microscope, or even a question that just asks about why the electron microscope was important. And one of the things the electron microscope has done is improved our understanding of the role of subcellular structures. So there have been developments in our microscope technology. And electron microscopes, because they magnify so much more than a light microscope, they provide greater magnification and greater resolution. Now the definition of both words will come up in a slide after this, in a few slides, but this is key part of the learning. In what way is an electron microscope, so just a question for you to think about, in what way is an electron microscope better than a light microscope? It provides greater magnification and higher resolution. Improved resolution enables us to see small structures inside organelles. So what are the difference between an electron microscope and a light microscope? So like I said, common question. It can be a 3-mark question, a 4-mark question. It is more likely to come in a foundation paper, but it can still come in a higher paper too. So the problem with the electron microscope is it's large and it's not portable. It's much more expensive. It provides higher magnification and resolution for detailed images. It's unable to view live specimens. So these are negatives. That's a positive, that's a negative. It does mean whatever they're looking at through an electron microscope needs to be dead. Light microscopes are small and portable, which is why we use them in schools. Lower magnification and resolution. It's much easier to use, and you are able to view live specimens. So from a school perspective, from a financial and other reasons, it's easier to use a light microscope. But the electron microscope has helped us to understand subcellular activity on a much better level. Now, this is an actual GCSE style question. What are the key differences between electron microscopes and light microscopes? So it's just what we've already covered. Electron microscope is a high resolution imaging device that uses a beam of electrons instead of light to magnify objects, allowing for detailed visualization of extremely small structures. So it gives better magnification, better resolution, and it uses electrons instead of light. A light microscope is an optical instrument, meaning it uses light and a series of lenses to magnify small objects such as cells and tissues. So why are electron microscopes more advantageous or better for viewing cells? So number one, it's got a higher resolution, meaning if you had, for example, 2 dots, now the gap between those dots is 1 centimeter, roughly 1 centimeter. I think that's, half a centimeter. Now our eyes can see the gap between those dots. Now electron microscope can see a gap of 0.1 nanometers. So that's a 0 followed by 10 more zeros and then a 1 meter gap. And that allows scientists to see much finer details within cells, such as organelles and macromolecules. So macromolecule is a large molecule, but using electron microscope you can see within that macromolecule. And it's better magnification. It can magnify up to 2 million times, whereas light microscopes typically max out at 1500 times. So key terms: resolution and magnification. So resolution is the ability to distinguish between 2 separate points in an image. So I explained on the previous slide. That means you get a sharper image. Magnification is the ability to enlarge an image compared to the actual object. So it's enlarging the image compared to its original. Now, magnification, and this is a very common question that comes in both higher and foundation, is calculation of magnification or size of image or size of real object. Now I always teach my students to remember this as I am, and I'll explain that on the slide on this slide. So magnification equals size of image divided by size of real object. So if we say let's rearrange this. So size of image equals size of real object times magnification, because size of real object is here and it's dividing. So if we move that to the other side, it multiplies. Now if we say size of image is an I, size of the real object, in other words the actual size of it, if we say that's A, and if we say magnification is M, what you've got there is I am. It's just an easier way of remembering it. Now let's have a quick look at this question. Now for anyone that's just logged in and just joined us, we are running about 10 minutes behind. So I am going to continue this session till 11:05. Between 5 past and 10 past, and then we'll have a 5-minute break, and then we'll start chemistry. So microscope technology. A bacterial cell is 0.04 millimeters long. So what we've got to think here is that the actual length that we're being given, is that the image length, is that the magnification. Calculate the length of the image after this cell has been magnified 500 times. So 0.04 millimeters, this is the actual. It's been magnified 500 times, that's the magnification. And what we're looking for? The image. So we're looking for I. So step 1, identify what you've got. So we know A equals 0.04, magnification is times 500. And in step 2, substitute the numbers in to the formula and then calculate. So 0.04 times 500 equals 20, and then make sure you've added the units. Now in higher, you may be asked to convert one unit into another. Now if anyone's struggling with converting units, if you let me know in the in the chat, then what I'll do is I'll go through it during one of the question answer sessions. Now this is specific to separate science only. So what I'm going to do is I'm going to go through this for separate science. So if anyone's doing combined science, if you want to take a break now, and we're going to restart at 10 past 11. For those doing separate science, I'm going to go through this and then we'll start the break. So culturing organisms. So this is the process for the aseptic technique for culturing bacteria. So number 1, sterilize the petri dish and agar to eliminate any unwanted microorganisms. Step 2, heat the inoculating loop in a Bunsen burner flame to ensure it is free from contamination. Step 3, so initially we've got a petri dish, which can be seen at the bottom. We add our agar to it, but before we add anything, we have to sterilize it. That way there's no microorganisms, because the whole point here is we're trying to culture a bacteria, we're trying to grow bacteria. So what we don't want is another bacteria contaminating it. Now, when we add our specific type of bacteria, we add it with an inoculating loop, which is this kind of ring of metal. Now you can use that using a Bunsen burner flame. Now then you take the sterilized inoculating loop. Once it's cooled, that is, you transfer bacteria onto the agar plate. So you would just scratch along the surface. Then you secure it with a lid with tape. So you put the lid over the top, you put the tape around it, and place the petri dish upside down. That prevents condensation from dripping onto the culture. Because that way, because it's upside down, none of the water because the water can hit the top of the lid, and start dropping down, almost like a mini rainfall within the Petri dish. And then incubate at 25 degrees. It's important to incubate at 25 degrees because our body is at 37 degrees. So if we start incubating at 37 degrees, the kind of bacteria that might grow are the ones that are harmful to us. Okay, so let's quickly look at the procedure, one step at a time again, but look at the justification between each one. Now I did justify it in the previous slide, but it is always better to read through it a second time. So why do we sterilize the petri dish and the agar? Because it prevents contamination from unwanted microorganisms. Why do we sterilize the inoculating loop? Because it ensures only the intended bacteria are transferred. Why do we use the inoculating loop to transfer bacteria onto the agar plate? Because it helps in controlled bacterial growth. It ensures that we're putting the bacteria onto the actual agar. Why do we seal the petri dish with tape? It prevents contamination and it stops condensation from affecting bacterial growth. And why do we incubate at 25 degrees? Because it encourages bacterial growth while reducing the risk of harmful pathogens. Okay, thank you everyone. If we're going to take a 4-minute break and we'll restart at 10 minutes past 11. Thank you everyone. See you in 4 minutes. Okay, welcome back. So we're going to start now, or we're going to continue with Chemistry Paper 1. Thank you for all the questions, and our team will keep answering as we're working through. Now with Biology Topic 1, we have fallen just slightly behind because we started a little bit late, but that's not a problem, we'll catch that up next session. So let's quickly put this on presentation mode and then get started. Okay, now just in case anyone missed the start of the last session, I'm just going to replay the video that explains who we are and what AMALearning stands for.
Providing you with the equation for success. Activate plus memorize plus apply equals learning. AMALearning, you're about to.
Okay, apologies, I'm just going to put the presentation back up. Okay, so as I explained before, with AMALearning, the A stands for activate, so that's understanding, and that's what we're doing through these sessions. So the AMALathon 1 is all about activating the brain. It's understanding every single spec point, and what you can do during the whole process is, if you don't understand a specific specification point, you can ask me a question, you can watch one of our videos, you can ask one of your teachers, whatever's necessary. But the whole point is you've now understood where your gaps are. Now, in the past, especially for example, when I first became a teacher, a lot of the feedback that was given to students was quite generic. So, and students will leave after doing an assessment and say, oh, I got the question wrong on the topic of periodic table. And they'll go home and they'll start learning the whole topic, when in reality they probably understood 90% of it, and it was only that 10% that needed targeting. So what they actually did was they used up their valuable revision time in the wrong way. What the AMAL process does is, because it's linked to specification points, when you're activating your brain, you're understanding everything. Because we're going through it one spec point at a time, you can identify where your gap is, and that helps with using your time efficiently. It's going to help, it's going to give you more time for memorize, it's going to give you more time to actually apply. And I know some of you missed this, there was a question that I'm just going to very quickly answer. After AMALathon 1, we'll have AMALathon 2. But AMALathon 2 is made up of two types of learning. So one is the activate phase learning, which will be in the May holidays. So in the May holidays, just like what I'm doing now, I will cover all of paper 2 content. And prior to every single exam, I'll be doing a masterclass. It'll be a past paper based masterclass. In a sense, it'll be like a walking, talking mock. And it will be also like a predicted paper. So based on my analysis of all the exams since 2018, I'll be going through all the topics which I think are most likely to come up. Now, usually I'm around 80% correct. And students that listen to me go away and learn what I've asked them to learn usually get grade 8s and 9s. So I do know what I'm doing. Our team knows what they're doing. So if you want to join us for that, there is the registration for that has opened. If you go to our website, the website link is below. One of the best ways for you to get information actually is to join our WhatsApp group. And if you want the resources for today, you can get that through our WhatsApp group as well. And the link to the WhatsApp group is below in our posts. Now, so for example, if we have an assessment, you've all got Biology Paper 1 on the 13th of May. On the 10th of May, I'll be spending 3 hours going through loads of past paper practice, based on our analysis, just getting you ready for the exam. Now this is our structure for the week. So we're going through atomic structure today. Now, so we'll go through atomic structure today. Tomorrow we're going to go through periodic table. Now, I think I should be able to finish atomic structure and finish most of periodic table today, hopefully. And that will give me a bit of time to catch up the biology, because of our 10-15 minute delay in our start, we fell a little bit behind. So it's possible we'll do a little bit of biology at 11 a.m. tomorrow. Like I said when I initially started, this is a rough roadmap. This is what we're going to try and stick to. But if we need to give additional time, we'll use this 9 a.m. time to catch up. That 9 a.m. time we've actually specifically put for those people that are doing separate science. So if there's anything that comes up, but it can be used for combined science too. So if anything comes up in anything we're doing right now, ask the question. Just say, I didn't fully understand that topic. I'd like to learn a bit more about it, or I'd like to go into more detail. And what we'll do is we'll use these timings from tomorrow for that. If it's something I can do in the question and answer session, I'll do it in the question and answer session. If it's something that I can give extra time on, we'll put out a post, and we'll put it into our WhatsApp channel, which is why I'm saying the WhatsApp channel is really important. And we'll let you know, actually, we're going to start from 9 and we're going to tackle that, give a bit more time to it. Yeah, Amal is here for you. We want you to leave our session saying, AMALearning, we just did. So we'll do whatever we can to help and support you to get to that point. So 1.1, atoms, elements and compounds. Now I know a lot of people will find this topic quite straightforward, but I'm going to go through it in detail anyway, because it's a foundation topic. By foundation, I don't mean it's a foundation topic and it'll only come in a foundation paper. I mean it's a topic that feeds into everything. If you understand this part of chemistry properly, all of topic 1 properly, you will understand electrolysis better, redox better, all the topics that some of us are struggling with. So how has the atomic model changed over time? All substances are made from tiny particles called atoms. So there's just a question I'm just going to quickly answer for those that were doing biology. So when we covered biology, with the separate science students, the culturing bacteria topic, which was 1.1.6, it is a required practical. Now, with the required practicals, you can be asked, sometimes you can get questions that cover, which are just on that required practical. Usually out of 100 marks out of the paper, you're expecting at least 10 to 15% to be linked to a required practical. So it's important to do lots of past paper practice. Now, for required practicals, I will go through as much of the theory and content as I can, but what I would advise is watching one of the exam board based practical YouTube videos, because they've made that video specific to that core practical, to that required practical. So because I can't do any required practicals live, I would advise watching a YouTube clip on it, but I will still go through the theory. Apologies. So to continue now, all substances are made from tiny particles called atoms. An atom is the smallest part of an element that can exist on its own. What are elements and how are they represented? So the elements are represented on the periodic table. An element, and each element has a symbol made up of 1 or 2 letters. The first letter is always capital. There are nearly 100 different elements. The elements are arranged in the periodic table. Now, while I've got the periodic table here, I'm going to cover something which will be explained in more detail after. If you go across the rows, so for example, if you look at hydrogen and helium, they're in row 1. So we call that period 1. Lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon are in row 2. So we call that period 2. Sodium, magnesium, aluminium, silicon, phosphorus, and so on are in period 3. Now, depending on what period it is in, that represents how many electron shells that particular element has. So for example, sodium is in period 3. So sodium automatically has 1, 2, 3 electron shells. Cesium, actually I'm not going to be able to draw 6 shells. Potassium is in is in period 4. So it will have 1, 2, 3, and 4 electron shells. Now, if we look at the columns, this is the first column, so we call it group 1. This is the second column, so we call it group 2. Boron and aluminium is in group 3. Carbon, silicon are in group 4, and so on. So we've got group 5, group 6, group 7, and then helium is in, we either call it group 0 or group 8. What the groups tell us is how many electrons they have in the outside shell. So if we look at sodium again, sodium was in period 3, so it's got 1, 2, 3 shells. But it's in group 1. That means in the outside shell it's got 1 electron. Potassium is in period 4, so it's got 1, so this is potassium here. It's got 1, 2, 3, 4 shells. Why has it got 4 electron shells? Because it's in period 4. But it's also in group 1. So how many electrons in the outside shell? 1. So let's pick a random one. Let's look at beryllium. It's in period 2. So if it's in period 2, it's got how many electron shells? 1, 2. But it's also in group 2. So how many electrons in the outside shell? 1, 2. I'm going to erase this one here. And let's have one more go. So if something makes sense, or if I've helped you in any way, just if you can give me some, if you're able to make a comment, that would help me as well, so I can see what people haven't understood. But if something does definitely come up that hasn't made sense, please let me know. I'm happy to explain something a second time round. So now let's look at aluminium. No, sorry, not aluminium. Let's look at sulfur. Now sulfur, first thing, it's in period 3. 1, 2, 3. So it's going to have how many shells? 1, 2, and 3. It's also in group 6. If it's in group 6, how many electrons in the outside shell? 6. So 1, 2, 3, 4, 5, and 6. Excellent. So what we've actually covered is a lot of the slides that are about to come, but I want to explain it while you can see the periodic table in front of you in my own words. So how are compounds formed and what happens in chemical reactions? So compounds are made by combining elements in chemical reactions. Chemical reactions always make new substances, and we can change energy that we can measure. How can compounds be represented and how are they formed? So compounds like water are made of 2 or more elements that are chemically joined in fixed amounts. So here you can see we've got 1 oxygen and we've got 2 hydrogens. So if we've got 2 hydrogens, that's H2, and we've got an O after it, so that's H2O. Compounds can be used using formula that uses the symbols of the elements that make them. To separate compounds into elements, you need to use chemical reactions. So in a sense, what the formula tells us is the ratio, well, not in a sense, it tells us the ratio of each atom within that compound. And each atom each element is attached to each other with a chemical bond. And we'll cover bonding in topic 2. So that will be in Monday's session. Now, for those who are only joining us for Saturday and Sunday, because their schools don't break up, because I know the schools from Birmingham aren't going to break up to the following week. Everything's recorded, so you can come back to the sessions. Moving onwards, however, we'll only be doing sessions at points where everyone is off. So it'll be common holidays. For example, the 1 week off in May will be a common holiday, and other sessions will be on Saturday or Sunday prior to each exam. Now, a quick thing to just explain here also is this. If I write H2O, it means 2 hydrogens attached to oxygen. If I now put here 3 H2O, that means I've now got 3 of these. So what that actually means is I've got 1, and then I've got a second one, and I've got a third one. So it is more a refer that the larger number at the beginning refers to how many of those molecules we have, whereas the superscript number refers to how many of that individual element we have. So that 2 refers to how many of that specific hydrogen we have. So what is a mixture and how can it be separated? A mixture is made of 2 or more elements or compounds that are not chemically joined. So here we've got an element. Here we've got an element, but it's a molecule, because it's still an element because there's only one type of atom there. Here we have a compound. So we've got 3 different types of molecules. We've got a single atom element here. Here we've got a molecule, but we're still an element. And here we've got a compound. And they share the same space. They're not chemically attached to each other. So there's nothing attaching this to this. So therefore it's a mixture. And what that means is they can be separated. Because they're not attached to each other, you can separate them. The chemical properties of each substance in the mixture stay the same. Meaning, this element here has its own properties. This elemental molecule has its own properties. And this compound has its own properties. Mixtures can be separated using physical methods. So these physical methods do not involve chemical reactions. Because if you had a chemical reaction, then you'd start breaking bonds and remaking bonds. Then things would attach to each other. For example, if you had a hydrogen molecule and you had an oxygen molecule, they'll start reacting with each other to make molecules of water. So when you separate a mixture, it has to be physical. Because if you separate it using a chemical method, then you can end up creating or making new substances. So how does chromatography separate mixtures, and what are the 2 phases involved? So insoluble substances are removed by the filter paper. So a good example here would be if you've got a beaker, if you've put into this a mixture of water, you've got some sand in here, and you've got some salt. The salt will dissolve into the water, but the sand doesn't. So when you add this mixture into this apparatus, the filter paper will catch all of the sand, because the sand's insoluble and it's too big to fit through the porous. So by porous I mean gaps, it's got tiny little holes. This filter paper has got little holes in between that allows the water to go through. So the water will go through and then collect as a filter at the bottom, but the sand will be captured by the filter paper. So substances that can dissolve pass through the filter paper. And then we've got another example. Now this says chromatography, so that should not say chromatography. This is crystallization or evaporation. So this method involves heating the solution to evaporate the liquid, reducing the volume and then leaving it to cool. So what you're actually doing is, initially the whole thing would be full of water, but initially you would boil off a lot of the water so that what you're left with is just a small amount of water that's saturated with whatever salt has been dissolved into it. Then it's left on, for example, a windowsill, or it can be put into a water bath because you can control the temperature in a water bath. Now the remaining water will diffuse, will evaporate and diffuse away, and what you're left with then is crystals of that salt. Okay, it says chromatography again. This is distillation. So this is simple distillation, and what you've got in the mixture initially is a liquid, and this can be two liquids, or it can be again like a salt solution. So you've got some salt in here that's dissolved in, or you've got two liquids. The liquid with the lower boiling point will evaporate, make its way down this small tube. Now you can see that this tube's got this cooling jacket around it. What will happen is cold water will flow through this, and as that cold water flows on the outside of the tube that the gas is going through, it helps condense it. So we've got two steps here. We evaporate the liquid, and then it says cooling there, but the better would be condense. We condense it back to a liquid. And what you're left with here is the liquid with the higher boiling point, and what you've got here on in the beaker is the liquid with the lower boiling point. Or it could be that you leave the solid behind here, and you're collecting the liquid on this side. Okay, this one is now fractional distillation. When we go through organic chemistry, I will go through this in a lot more detail. But what we've got here is a temperature gradient. So it's hotter at the bottom and it's cooler at the top. So what you would do here is you get a mixture. Now this is specific to crude oil, but it could be used for any mixture, I suppose. But if you had crude oil, which is a mixture of hydrocarbons, and because it's a mixture, depending on how big the carbon chain is within the alkane, within the crude oil, because you've got a longer carbon chain, it will have a higher melting and boiling point. The ones with short chains will have lower melting and boiling points. So all of crude oil is heated up, turned to a gas. At that point, still there's something called bitumen, which is a liquid, and that flows off at the bottom. But the remaining part of it, I'm just going to change the color of my pen so you can see this more clearly. So the rest of it enters as a gas. So the rest of it enters as a gas. That gas starts diffusing upwards, but as it does, when it hits different temperatures, some of it starts to condense. So for example, at 250 degrees, where my blue line is at the moment, diesel condenses, turns to liquid, and it can be siphoned off. But the rest of it's still a gas. So that gas still keeps moving upwards. And at each point, so for example, when we reach around 100 degrees, naphtha turns into a liquid, and that gets siphoned off. Around 60 degrees, petrol on 66 degrees, petrol turns into a liquid, so it condenses and is siphoned off. So again, we're boiling it or evaporating it, so it turns into a gas, and then we're condensing it. So distillation always has these two steps. And this actually is chromatography, so that is correct. This method involves heating and cooling a substance to separate it based on its boiling point. So this method involves heating and cooling a substance to separate on its boiling point. No. So what, again, this will be covered in more detail afterwards, but what is done with the chromatography paper is initially you draw an origin line. So on your chromatogram, you draw a line in pencil. Pencil, because it doesn't dissolve into the water, it's insoluble. Then you take your chemical that you're testing, you put a dot of it, a small amount of that onto that pencil line. You now add it to the water. You make sure your water line is below that pencil line. Now what will start to happen is the water will start to be absorbed by the paper and climb through the paper. So as it starts to move up the paper, called this capillary action, it will get to a certain point. So for example, the water has now gone up to here. Now as it goes past the sample that we've put, the dot of sample that we put on there, that will dissolve into the water and that will start moving upwards too. So for example, and it will split into its original colours that it's made up from. So for example, this one might have been made of 3 different colours. Now what you can do is you can take a ruler and measure how much that particular colour has moved. So that might be, for example, 3 centimeters. And with the ruler you measure how much the water's moved, and that might be 10 centimeters. Divide those two numbers. In this case you get 0.3, and that's what we call the RF value. Like I said, we are going to cover this in more detail. It comes up again, but I just want to give you a brief understanding of it now. And what that helps us to do is identify pigments within ink, or it helps us to find out what something is actually made up of. If it's a pure substance or pure color, or is it made up of lots of different things. So developing the model of the atom. This is something that we'll cover again in topic 4, physics. So when we start doing radiation, this is the starting point of that topic. So at that point, I won't go through it in as much detail unless I'm asked to. So ideas about the model of the atom have changed over time. This happens because new experiments give us new evidence, so scientists update or replace models. So Dalton, he suggested that atoms were tiny spheres that could not be divided. Electrons at that point had not been discovered yet, but there was an understanding that came from the Greeks that, and it was an understanding that came from the Greeks that at the moment, we might not understand, though they didn't understand what was in the atom, but they did understand that if you keep dividing something over and over again, then eventually you can get to something that can't be divided. And Dalton expanded that a little bit. Dalton suggested that the atoms were tiny spheres that could not be divided. So similar to what the Greeks said, but he expanded on it a little bit further. Now, over time, new evidence has come into place, and so our understanding of the atom has changed. For example, J.J. Thomson discovered the electron. So he suggested the plum pudding model. And what his suggestion was that Oops, sorry. Right, apologies. Just be 1 second getting the presentation back up. So what he suggested was the plum pudding model. And what he meant by that was, because at that point they didn't understand that there was electron shells, his new discovery was he assumed this circle, the small circles were negative, and they were negative. So these small, I'm shading them in blue, were negative, and the bit around it was all positive. So he said that the atom is a ball of positive charge. So the bit I'm shading in now is the ball of positive charge, surrounded by with tiny negative electrons stuck inside it. Now, we don't plum pudding is not something which is that common now. So you could look at it as a chocolate chip cookie, where the cookie is the positive part and the chips are the negative. Now, then came Rutherford and the alpha particle scattering experiment. This is the experiment that massively changed how we understood everything. Now, if we consider the atom and we look at it from a John Dalton perspective, then everyone assumed it was just a solid sphere. Now, if you have a solid sphere, then if you fire a particle at it, and the particle they fired at it was an alpha particle. An alpha particle is made up of 2 protons, which have a charge, and 2 neutrons. Now, that is the same as the helium nucleus. Now, when we say helium nucleus, what we mean is the helium atom, but without the electrons. So that's what an alpha particle is as well. So we're going to cover this in topic 4 later on in the week. But it's 2 protons, 2 neutrons, and that's the helium nucleus. So that's the atom without the electrons there. Now, if you fire that alpha particle, you'd assume that it would just bounce back. But what they actually found was the majority of it just went straight through. And what that indicated to them was that showed that most of the atom must be empty space. So if we're now looking at the atom as just a circle, they assumed that actually most of it's empty space now. Now, every once in a while, in fact 1 in 18,000 alpha particles fired would hit something and bounce right back. And slightly more, but not many, as they would go in a straight line, but then they would get deflected and collide with the side. So they then assumed the fact that something's bouncing back, there must be some subatomic particles in the center. So this showed that the atom has a tiny heavy center. That's what the alpha particle bouncing back off. The fact that it was getting deflected. Now, what's the charge of an alpha particle? It's positive. If it's bouncing back and if it's being deflected, what does that suggest? And think of this from a magnet perspective. If you've got 2 magnets, you've got the plus side of a magnet here and a plus side of a magnet here. What do? They deflect. They push each other away. They repel each other. And that's what essentially was happening. Whatever was there was repelling this positive charge. That means it must also be positive. So therefore they concluded that this nucleus must be positive. Now they knew that there was electrons, so they assumed the electrons must be in like an electron cloud on the outside of the atom. So we went from, so the alpha scattering experiment showed that an atom's mass is in the tiny central nucleus, which has a charge. This led to the nuclear model replacing the plum pudding model. So what I just drew was the nuclear model. So let me just recap that. Because lots of alpha particles were going straight through, they concluded there must be a massive empty space. Because 1 in 18,000 or so alpha particles bounce back, they concluded there must be a heavy nucleus in the middle. And because some of the alpha particles are being deflected and the alpha particle is positive, they concluded there must be a positive nucleus. So we went from our plum pudding model to our nuclear model, which is similar to what we consider it to look like now. So ideas about the atom have changed over time. So this is just a recap of that. Rutherford did an experiment called the alpha particle scattering experiment. From this, he suggested the nuclear model of the atom. Now, Niels Bohr improved that model. Because what Niels Bohr did, he suggested that electrons move around the nucleus in fixed orbits at specific distance. So that was based on his understanding, and that was based on his research. So based on his understanding, he understood that there must be electron shells, and the electrons must be within those electron shells. Just a quick answer to a question: if anyone is looking for the resources, all you've got to do is join our WhatsApp channel and you can find the resources there. We've just found it to be an easier way to pass on resources than sending emails. Now, James Chadwick, when he started to weigh the masses of atoms, he realized something wasn't making sense, that there must be something else within the nucleus, and then he discovered neutrons. Now, questions like this do come up. What most questions do is ask you to describe the difference between what John Dalton believed and what Rutherford believed, as in how things changed. You might get asked questions about Rutherford's experiments. You might get asked just a really simple question, as in what did Niels Bohr discover, or what did James Chadwick discover, or the other way around, who discovered neutrons, etc. Small one-mark questions that do come up. But usually when this topic comes up, it's usually a comparative. So, why are atoms neutral? In an atom, the number of electrons is the same as the number of protons and neutrons. So, let's look at it this way. The charge of a proton is positive. The charge of an electron is negative. So if we look at, for example, let's look at carbon. Now, what do we call that bottom number? That is our atomic number. That gives us the number of protons, which in this case is 6. That bottom number also tells us the number of electrons within an atom. Now, when we look at ions afterwards, ions always have a different number of electrons, but atoms have the same number of electrons. So if there's 6 protons, there's going to be 6 electrons. Now, just to finish this example off, how many neutrons, what does the top number tell us? It tells us protons and neutrons. So if we take away these two numbers from each other, what we're left with is just neutrons. So 12 take away 6, 6 neutrons. So let's still keep looking at carbon. If I've got 6 protons, if each proton is a +1 charge, that means we have a charge of 6 plus. Now if we look at the electrons, if I've got 6 of them, each one has a -1 charge. That means the charge overall is going to be 6 minus. So what's my overall charge? 0. So the overall charge of every single atom is 0, it's neutral. And neutrons have an overall charge of 0 anyway. So this means the atom has no overall charge. Because this is what we just covered on the previous slide. Okay, size and mass of atoms. So atoms are extremely small. This is a very rare question about that, and you sometimes do get questions about how small an atom is, or how small, or a comparative question between the radius and the atom. But usually they give you a size, and it's just a mathematical question where you would divide the radius of the atom by the radius of a nucleus. I have seen a question which said, how much bigger is the atom when compared to the nucleus? And all you have to do is divide that number by that number. And they provided both numbers. They're not expecting you to memorize such numbers. But the understanding here is that the nucleus is much, much, much smaller. In fact, 1/10,000th of the actual atom itself. I've seen this come up in a foundation question, where it's a multiple choice question. Now, nearly all the atom's mass is found in the nucleus, because the proton has a relative mass of 1, and the neutron has a relative mass of 1. The electron, I know it says very small and almost 0, but the better number is to say is 1/1836. So let me write that again because it's not come out very clear. 1/836, meaning the proton and the neutron weigh 1,836 times more than the electron. So, and that again goes alongside what Rutherford explained, that you've got the majority of the mass in the central point. Now, what does each tile on the periodic table actually tell us? So, as I explained before, if we look at carbon as an example, well, let's look at oxygen this time. The bottom number tells us number of protons, and the top number tells us protons plus neutrons. So the bottom number tells us protons, so we've got 8 protons. Within an atom, the number of electrons are always the same. And how do we find out neutrons? We deduct top number minus the bottom number. So 16 take away 8 is 8. Now, these 3 numbers are not always the same. It's just because of the examples I've used. Now, an isotope, I'm going to give a quick example of, just in case anyone's in the middle of an exam and they forget what an isotope actually is. So if we look at, for example, carbon-12. Carbon-12 has got 6 protons and it's got 6 neutrons. 12 take away 6 is 6. Now if we look at one of its isotopes, and we're going to say the definition in just a second. It has 14 mass number. So this is carbon-14, and it has a proton number of 6. Now the fact that they've both got an atomic number, a proton number of 6, means they are both carbon. They're both the same element. Now in this case, we have the same number of protons, which is 6. 14 take away 6 is 8. So the number of neutrons is 8. Meaning we have the same number of protons, but we have the different number of neutrons. And that's what the definition of an isotope is. So it's something that is the same element. They're both carbon. How do we know they're the same element? They have the same number of protons. But how do we know they're isotopes? They have a different number of neutrons. So I'm just going to delete some of that so you can see the writing underneath. Atoms that have the same number of protons but different number of neutrons. So for that example, if you ever forget in the middle of an exam, just quickly draw out that example. So you can see that they've got same number of protons but different number of neutrons. Relative atomic mass. So this is the average mass of the element's atoms, taking into account the different isotopes and their abundance. Now, this does come up in more detail afterwards, but I'm just going to explain using chlorine as an example. So chlorine, if you look at your periodic table, strangely enough, the top number is 35.5. But in reality, there's the two main isotopes of chlorine are chlorine 35 and chlorine 37. Now 75% of chlorine is chlorine 35 and 25% of chlorine is chlorine 37. So if we do a quick calculation, so we find out 75%, so when we say percent we mean out of 100, so 75% times 35, and to that we add 25%. So I should point out in the past I have held senior leader positions in schools, but I've also been a head of maths, a head of science, which is why when I'm teaching maths I do teach it the same way I would teach a maths lesson. So we're saying percent, we're saying out of 100, so we find 25% of 37, so we're finding out 75% of 35, 25% of 37, and that equals 35.5. So that's why we call it relative atomic mass. So it's relative to all the different isotopes. So on your periodic table, this is why you see 35.5, and because what a lot of people commonly ask is, how can you get half a proton or half a neutron? In this case, half of a neutron. It doesn't make sense. But it's because it's an average between the isotopes. So here it says the average mass of an element's atoms, taking into account the different isotopes and their abundance. Okay, electronic structure. So in the first electron shell, we always get 2 electrons. How many do we get in the second shell? 8. And how many in the third shell? 8 as well. And for the time being at GCSE, you can put the remaining electrons into the fourth shell. But when you do A levels, it'll go into a lot more detail. It'll talk about the suborbitals. And in this, the example that I showed, we've got 2 electrons in the first shell, we've got 8 in the second shell, and we've got 8 in the third shell. So that's its electronic configuration. Now if we look at oxygen, because that bottom number is 8, oxygen has 8 electrons, which mean 2 electrons go in the first shell. So from our 8 electrons, we've now got 6 left. What's the maximum we can put in the second shell? We can put 8. So we've only got 6 electrons left, so we're going to put all of them into the second shell. So our electronic configuration, how many electrons in the first shell? 2. How many in the second shell? 6. So the electronic configuration of oxygen is 6. The electronic configuration of argon is 288. Now, this initial part of 1.2.1 is what we covered at the beginning of this lesson. and that is, when you go across the period, what does the period tell us? It tells us the number of electron shells. So that means hydrogen and helium only have one shell. This is period 2. So that means lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, and neon will all have 2 electron shells. And for example, francium, which is in period 7, will have 7 electron shells. So 1, 2, 3, 4, 5, 6, and 7. And now if we look at all of these, what period are lithium, beryllium, fluorine, and neon within? They're all period 2. And how do we know that? They've got 2 electron shells. Now, if they're in the same group, for example, lithium, sodium, and potassium are in the same group, what does that tell us? It tells us that they have the same number of electrons in the outer shell. So lithium has 1 electron in its outer shell. Sodium has 1 electron in its outer shell. Potassium has 1 electron in its outer shell, so they are all in group 1. They're all in group 1. But from a period perspective, what period is lithium in? It's got 2 shells, so it's in period 2. What period is sodium in? 1, 2, 3 shells, so it's in period 3. What period is potassium in? 1, 2, 3. It's got 4 shells, so it's in period 4. But they've all got 1 electron in the outer shell, so they are all in group 1. So again, what group is fluorine in and what group is chlorine in? So if we count the electrons, we've got 1, 2, 3, 4, 5, 6, 7. So that's in group 7. Chlorine's got 1, 2, 3, 4, 5, 6, 7 electrons in the outer shell, so it's also in group 7. But from a period perspective, fluorine's got 2 shells, so it's in period 2. Chlorine's got 1, 2, and 3 shells, so it's in period 3. Now, group 0 means that they have full outer electron shells. So remember, if you think back to the slide on electronic configuration, what's the maximum number of electrons in the first shell? It is 2 electrons. That's why helium has a full outer electron shell, because it can only have 2 electrons. Neon has got 8 in its second shell. What's the maximum number of electrons we can have in the second shell? It is 8. Argon's got a full outer electron shell, because what's the maximum number of electrons we can have in the third outer shell? It is again 8. So because they all have full outer electron shells, we call that group 0. Okay, from tomorrow we're going to continue from how the periodic table was developed. So we'll be looking at Mendeleev and how we went from Mendeleev's table to the modern periodic table. Okay, thank you everyone. So we are going to continue at 10 past 12. So like I said, we are about 5 minutes behind now. So when we restart at 10 past 12, we will continue till 5 past 1. Okay, thank you everyone. I'll see you all in 5 minutes. Okay, welcome back. We're now moving on to the physics part, the final hour of our session. Tomorrow I'll be on from 9:45. So if anyone does have any questions, they can just come on live and ask their questions at that point. I have at the moment been answering live. I think we have answered all the questions. Either someone from the AMALearning team has answered live within the live chat, and I've answered a few questions as well. But if you want to log in at 9:45, I'll be on, and you can ask one-to-one, and I can show you help answer your question. Now, moving on to physics. Okay, we have seen this twice, so I won't play that again. But with our physics, what we're looking to do in today's lesson is we're going to start on the topic of energy. Energy will most likely take till tomorrow to finish. Then move on to electricity. Electricity being one of those topics that I know a lot of students struggle with. And then we'll move on to the particle model. And by the end of the week, we will finish with atomic structure. And I have given over 2 hours to atomic structure. Because again, alongside electricity, it's one of the topics that students do struggle with. Just understanding radiation in a lot of detail. So we are now going to continue till 1:05, and then we'll finish there. And like I said, all the recordings are available, so you can watch the videos again and help you with the revision. Now, apologies, at the beginning we did have some issues when we started with biology, so I just want to apologize for that. It is a new process for us too, but we've got a really good understanding of it now, so it isn't something that will happen again. And one of the things that in that kind of rush, I forgot to introduce myself. So my name is Suhaib Tanvir, Mr. Tanvir, as I'm known to my students. I have been in education for over 15 years. And part of the reason we actually made AMALearning is because with our team, we've got a few different types of experts. We've got people who are experts within software engineering, with app design, myself from an educational background. And what we wanted to do was do like a joint project. And for a long time now, I've been achieving really, really good results. A lot of my students go from grade 2s and 3s all the way to grade 7s, 8s and 9s. Foundation students who are failing end up at least getting grade 5. Students who really want to go into medicine and dentistry that are struggling to get past that grade 7. And it was the methodology that I used, and what we found is the methodology that other teachers used was quite similar, which was the AMAL methodology that I mentioned before. That activate your brain, understand everything, and the way these PowerPoints are made help you identify where your gaps in learning are. In fact, our YouTube channel, if you look at the animations, there's an animation for every single specification point. But at the moment, it's just biology topic paper 1. But we're making those as quickly as we can. We have quite a lot of animators working on that. So anyone who's on here from year 10, by next year, you'll have access to a video for every single animation, an animation for every single specification point. And then the next 2 phases can be done interchangeably. It's apply what you've just understood to past paper questions. And what that does is it gives you an understanding of what examiners are looking for, and it also shows you what isn't being memorized. Now the activate phase is done through these kind of sessions. So we'll be doing this session again for all of paper 2 in the May half-term holiday. And the apply phase we'll be doing with pre-exam workshops. So if you really enjoyed or you're liking how I'm teaching, there are things that you can start registering for from now. I know the initial topics that we're covering are topics that you're probably really familiar with, but I still want to go through everything. So students have got a chance to ask questions about everything as well. Now, just again, because I was talking about myself, so I have been in education for 15 years. I've worked in senior leadership, particularly on results strategies. So the schools that I've worked in were schools that were struggling with results, and then I've helped improve whole school results. And I've worked as a director of maths, director of science. Currently, I'm a head of science. I'm director of science, but I have worked in lots of different positions. So hopefully I do know what I'm doing. But like I said, you can keep adding your comments. If you've got questions, please asking. If there's anything you want me to do differently, you can put that into the live chat. Okay, so let's get started. Conservation of energy. So first thing you've got to understand is what a system is. In physics, a system is a set of parts that exchange energy and matter with the surroundings. It can be open, closed, or isolated. Now, it's not as important to know the isolated part. So I'm just going to talk mainly about open and closed. Now, if you have an open system, both energy and matter can enter or leave. What that means is, if I've got a cup of hot cup of tea, it's going to cool down because the heat energy can leave. And it can radiate from the sides as well. Now, if I put it on a hot metal plate, or on a hot plate, then energy can also transfer inside as well. So that's what I mean by open system. Energy can go in, energy can come out. A closed system, so for example, a boiling kettle. A closed system, energy can transfer, but matter cannot enter or leave. So for example, a sealed thermos. So because it's sealed, so imagine if I seal this, you will still over time get a slight amount of heat loss, but now all the liquid that's inside is trapped within inside, so the matter can't leave that anymore. So you've cut off the movement of matter, because when that water is evaporating, it's causing water to leave. That's in an open system. In a closed system, the energy can transfer, but the matter can't go anywhere. Now, let's quickly go through the different energy stores and have a quick look at the description of each one. Because if you can understand this part here, you can answer a lot of questions. This is, again, it's a foundation topic. And by that, I don't mean a foundation topic. It only comes in foundation. It means it's a topic that will help you understand other topics. So magnetic, the energy store when attracting poles are pulled apart or repelling poles are pushed together. So magnetic is just the energy between magnets. Internal means the total kinetic and potential energy of particles in an object. So when we're talking about kinetic and potential, and I'll explain this in more detail when we go through the equations, but if I've got a ball, the higher I lift it up, the fact that it's moving, that's kinetic energy. But the higher up it goes, the more potential energy it has. When it drops, then that potential energy converts back into kinetic energy. So the higher up something is, the more potential energy it has. On ground level, because it's now not lifted off the ground, it has a potential energy of 0. Chemical, this is the energy stored in chemical bonds. So for example, if you've got a lump of coal, a lump of coal is made of carbon, and between all those carbons you have got bonds. So when you start burning it, what you're doing is you're breaking those bonds and causing a release of energy. Although the energy release is more when new bonds are made. Because when this reacts with oxygen in the surroundings, it makes carbon dioxide. When carbon dioxide is formed, it's an exothermic process, so it releases lots of heat energy. Then we've got kinetic, so that's the energy of a moving object. Gravitational potential, that's the energy stored in an object at height. Elastic potential, the energy stored when an object is stretched or compressed. So a bow and arrow is a good example, or a bubble or an elastic band. All of these are examples of that. Then we've got nuclear, the energy stored in the nucleus of an atom. Electrostatic, the energy stored when repelling charges have been moved closer together, or when attracting charges have been pulled further apart. So let's go through a few scenarios. When an object is projected upwards, As it moves up, that's kinetic energy. But as it's moving upwards, what's happening to the speed? It starts to slow down. And that's because the kinetic energy is decreasing because it's being converted into potential energy. So right at the top, you have a maximum potential energy. Then the ball starts to drop. And what does the potential energy convert back into? Kinetic energy. But this is just talking about the object projected upwards. So as it's going up, the kinetic energy is converting into potential energy. Now remember, potential energy, and this will come in a few more slides, to work it out is mass times gravity times height. In other words, if you increase the height of something, automatically what have you increased? The gravitational potential energy. Let's look at another scenario. Again, a roller coaster moving upwards. So as it goes up, however slow it's moving upwards, the kinetic energy is decreasing and potential energy is increasing. So, a moving object hitting an obstacle. So initially we've got kinetic energy because the object is moving. Now all these scenarios are subjective because each scenario could have lots of other energy changes as well. But once it collides into the wall, then we hear the sound, and as particles collide into each other, so for example, if you've got your hands and you rub your hands together, what happens to our hands? They start to warm up. So every time particles collide with another particle, we call it friction, and friction is happening all the time. So when air particles hit our face, or we're walking and you have a gush of wind, all of this is friction. But in reality, when you have friction, it releases thermal energy. So here, kinetic energy is converting into sound energy and thermal energy. So, an object accelerated by constant force. So the example we're using is for example, a car or a boat. What you put inside it is fuel. So you put petrol, diesel, some kind of fuel. And what that fuel then does, it burns and it releases energy, and that energy is used to make the machine move, being a boat or a motorbike or a car. And when it's moving, that is kinetic energy. So again, an object accelerated by a constant force. So when a person jumps out of a plane, as they're falling and they're accelerating, what's happening is the gravitational potential energy, because they're quite high up, they have a big height. As the height decreases, the potential energy is decreasing and it's converting into kinetic energy. So a vehicle slowing down. Again, you could, here it's moving quite fast. Then between the wheel and the brakes, you have friction. So that's a force. But as you've got friction, the particles are colliding with each other. What's being released? Heat energy. So thermal. So the kinetic energy is converting into thermal. As the kinetic energy decreases, the thermal energy increases. Now, kinetic energy equals, as a formula, and afterwards we'll cover this, half times the mass times the velocity squared. So if the kinetic energy is decreasing, you can't decrease the mass of something. What's the only thing that can decrease? So if kinetic energy decreases or Ke decreases, automatically the only thing that can decrease is the velocity, the speed. Hence, the object slows down. So another scenario, bringing water to the boil in an electric kettle. So you've got electric current flowing through it, and that converts to thermal energy. Now, what you do need to know is this formula and how to use it. And that's kinetic energy equals half times mass times velocity squared. Now in our notes that you can download or that will be provided to you, there's lots of practice questions that you can go through in your own time. I'm just going to go through a couple just as an example. Now, like I said, you can either log on tomorrow at 9:45 if you're struggling with these kind of questions, and I can go through it with you, or if you ask any questions in the live chat, I'll go through it in more detail. So here, the first thing you've got to understand, now in your exam this year, and if you're year 10 or year 9, this is going to come up again. You'll be provided with a formula booklet. So because you'll have a formula booklet that will have that full formula there, that does give you an advantage. However, you need to know what the units will be. So kinetic energy is measured in joules, capital J. Mass is always measured in kilograms, and velocity or speed is meters per second. That means if something is not in joules, kilogram, now they will always give you meters per second, so you don't have an issue with meters per second. You need to convert it first. So let's look at an example question. We've got a 750 gram book is thrown and has an initial speed of 12 meters per second. Calculate the kinetic energy. Now I'm going to do this myself at the side because before the answer actually comes up. Do we know what the kinetic energy is? So the best way to answer these questions is first write down what you know. Now kinetic energy, question mark. Do we know what the mass is? We know it's 750 grams, but we want mass to be in kilograms. So when you go from grams to kilograms, you're going from small unit to big unit. So that means you divide by 1,000. So if you divide 750 grams by 1,000, you get 0.75 kilograms. And we know that the speed is 12 meters per second. So the next step now is to substitute that into the formula. Just going to rub this out of the way. So we substitute, we know that mass is 0.75. We know V is 12. Now, at this point here, a lot of people forget the squared. So they miss the squared out and they get a completely different answer. So what we're doing here is half times 0.75, which equals 0.375, times 12 squared, which is 144. You don't have to do this in separate phases. You can now, at this point, put that whole thing through a calculator. Why can you put it through a calculator? Because in science, every single exam is a calculator paper. So always check your maths with a calculator. Do not try and do it mentally. There's no point, because you could accidentally make a mistake and lose vital marks. Now, half times 0.75 times 12 squared is 54 joules. Now, that could be an additional mark. So you need to know that kinetic energy is measured in joules. And 2 significant figures is enough in physics. So let's have a look at another question. We've got 1,300 kilograms. That's fine. That's mass. Here we've got an issue. We've got 1.2 kilojoules. So we're converting kilojoules to joules. So we're going from a big unit to a small unit. So when you go from a big unit to a small unit, you multiply. So if we multiply that by 1,000, it will equal 1,200. And we're working out the kinetic energy, and we've got the calculator working out the speed. Now, again, I'm going to write this all out so we can go through this one step at a time. So we know kinetic energy equals half times the mass times V squared. So let's just quickly add the numbers in and calculate it. So kinetic energy is 1,200 it equals half times 1300. So I'm going to just do that now. Half times, so that's half, and mass equals, actually I'll just write it down so everyone can see each step. 1300 times v squared. So that means we know that 1200 equals 650 times v squared. Now at this point, 650 is multiplying. So on the other side, if we look at it from an algebra perspective, it's going to divide. So now what we're going to do is v squared equals 1200 divided by 650. Now we want v to be the subject, so we want to do something with that squared sign. So what's the opposite of squared? So if again you look at an algebra perspective, what do we need to do to both sides? We need to square root it. So v equals the square root of 1200 divided by 650. So I'm going to rub this out. And then show you the answer that's already on the slide. Now, the big issue here with this particular question is that on the previous slide, I explained that people forget to square the velocity. In this question, people forget to square root. So they find out, they get to this point here, and they got v squared equals v squared equals 1.846, and they just put that as the answer. They just say v squared equals 1.85, and that's the answer. Now they'll still get 2 marks for that. If this was a 3-4 mark question, usually it's a 4 mark if it's got v squared involved. Then the final marks come from v equals the square root of 1.85, which is 1.36, which again to 2 significant figures is 1.4. So that means v equals 1.4, and you get a mark for the unit, meters per second. So what is the equation that you would use to calculate elastic potential energy? So with these questions, there's lots and lots of past papers. If you look through all the past papers, you're going to find a lot of kinetic energy style type questions. It is definitely worth practicing, because don't forget, in a physics paper, you're looking at least 30-40% of the paper is going to be maths based. So the more practice you do, the better. Now let's look at the equation for elastic potential energy. So elastic potential energy is half times K times extension or compression squared. Now K is a constant, or in this case the spring constant. So E, capital E, small e, that's the elastic potential energy. That's measured in joules. All energy is measured in joules. The spring constant is measured in newtons per meter. And the extension is measured in meters. So a spring with a spring constant of 2.5 newtons per meter is stretched by 18 centimeters. Calculate the elastic potential energy. So when we're converting centimeters to meters, we're going from a small unit to a big unit. So what do we need to do? In this case, divide by 100. So 0.18 meters. Now again, what a lot of people forget to do is they forget to square the extension. So if we look at the formula, it's half times spring constant times the extension squared. So that's 0.18 squared. So if we substitute the numbers in, half times the spring constant 2.5 times the extension squared, 0.18 squared, which equals 0.0405 joules. Well, we can round that up to 2 significant figures and we get 0.041 joules. and you get a mark for writing joules as well. Then the foundation paper, they most likely will have already given you the unit. In the higher paper, you're less likely to give the unit, unless it's at the beginning of the paper. Because the beginning of the paper is similar to the end of the foundation paper. So here's another example. A spring constant. Now if there's anyone that can answer this question faster than I'm going through it, you are more than welcome to have a quick go at it and give me your answer in the live chat. I know we've got a few people that are on today that are really fast at typing, so they're more than welcome to answer as much as they can. So first thing we do is converting the units. So like I said before, we're going from centimeters to meters, meaning we're going from a smaller unit to a bigger unit. So what we're going to do, we're going to divide by 100. So that's why we've got 0.25 meters. We're substituting the numbers in. Don't forget to square the extension, and hence the answer is 0.125 joules. And we simplify that to two significant figures, 0.13 joules. Okay, third formula now. We've just gone through kinetic energy, which was half times the mass times the velocity squared. We've just gone through elastic energy, which was half times spring constant times the extension squared. And now we are going through potential energy, which is mass times gravity times the height. And again, energy is measured in joules, mass is measured in kilograms. Now, with the gravitational field strength, you can't simply just say gravity. Now on Earth, gravity is 9.8. And whereas in an exam, usually they will give you gravity. They'll say G equals 9.8. If they don't give you G equals 9.8, you can just use the number 10. But if you can remember 9.8, that's the better number to use. Now, height is measured in meters. Let's look at an example question. A 350 gram book is lifted 2.8 meters when the gravitational field strength is 9.8. Calculate GPE. So again, we're converting from grams to kilograms. And that means we're going from a small unit to a larger unit. So what do we do when we go from a small unit to a large unit? We divide by 1,000. So when we go from small to big, you divide. When you go from big unit to small unit, you multiply. Okay, now these numbers that have been added here are slightly wrong, so I'm just going to add my own numbers in. But it is for a different question, so we can still look at that question as well afterwards. So what we will do is 350 divided by 1,000 is 0.350, so that's our mass. So that means if we substitute into this equation, we would do 0.35 times gravity, which is 9.8, times 2.8, which is the height it's been lifted. And the answer is around 10, but I'm sure the answer will come on the next slide, because I think what's happened is that possibly the two slides have mixed up. No, I don't think they have. And then you've got to make sure that your answer is given in that all your so you've got to make sure the answer is given in joules. Now we had a really good question on the live chat, which is it true we are given all equations in the exam? That's true. So for the next 2 years, you'll be given a formula sheet with all the equations for physics. And that's linked to the impact from COVID. They weren't going to, but there was a lot of negotiation, and thankfully they did agree to do that. Let's have a quick look at this question. So calculate the change in vertical height of a person sliding down a zip wire. So we've been told in the question, gravitational potential energy equals 1.47 kilojoules. The mass of the person is 60 kilograms, and the gravitational field strength is 9.8. So what we've got to do here is initially convert this. So this is in kilojoules, but we want it to be in joules, meaning we're going from a larger unit to a smaller unit. So what do? Because we're going from larger to smaller, we are multiplying by 1,000. So that's 1,470 joules. Substitute all this in. So we've got 1,470 equals 60 times 9.8 times the height. So now 60 times 9.8 gives us 588. Now on this side of the formula, it's multiplying. So what's it going to do on this side? It's going to divide. So we're going to do 1,470 equals 588 times the height. On this side it's multiplying, so on this side it will divide. So that gives us 1,470 divided by 588. And that gives us the height. Remember, height, the units for height is in meters. So the answer is 2.5 meters. That's been rounded up to 2 significant figures. So it's important that you round it up to 2 significant figures. 3 will still probably get you the marks, but it's okay to do up to 2. Now, when you get the equation sheet, it will not have units on there. That's why knowing the units is important. Because usually when you have, particularly in higher, if you're doing the higher paper, they'll give you the equation sheet, but they're not going to tell you what units it's in. So you might lose a mark. Particularly in the higher paper, you will lose marks for that. Okay, I'll leave this for you guys to go through in your own time. Okay, energy changes in systems. So what is specific heat capacity? Now, some of you will have covered this already as well. So specific heat capacity, going to the wrong slide, is the amount of energy required to raise the temperature of 1 kilogram of the substance by 1 degree Celsius. So what that means is, if you've got, for example, a piece of metal and it weighs 1 kilogram, is how much energy has to enter that. So you can use a jouleometer to work out how much energy is entering, how much energy has to go in that to take the temperature up by 1 degree. So if it started at 20 degrees, how much energy has just entered to take it up to 21 degrees. So this is the equipment that you would use to do this. So you've got your jouleometer, you've got your immersion heater, you've got your copper block, you've got a thermometer. The thermometer tells you the temperature. You've already used the mass pan balance. To tell you the mass, that's 1 kilogram. There's your copper block. That's the thing that you're checking to see what its specific heat capacity is. You've got a jouleometer attached to a heater. So now the heater starts to heat up the metal, and as it heats up the metal, it uses a certain amount of energy. The jouleometer will tell you the energy. So that's the amount of energy needed. As soon as it goes up by 1 degree, then you write down how much energy was used. So let me just recap that very quickly. You've got a jouleometer, which tells you the amount of energy used. That's attached to an immersion heater. The heater heats up this copper block, and when the temperature is going up by 1 degree, you write down you make a note of how much energy was used. So that's from this part here, from the jouleometer, and that gives you the specific heat capacity. So that gives you the amount of joules per kilogram per degree Celsius. So this unit here, that gives you joules per kilogram per degree Celsius. So again, this is an important formula, and you will very likely have to convert units. You'll have to rearrange. So this is where a lot of the math skills come in place. Look, just from this one topic, we've already covered 4 formulas that could come in the exam. So we're talking at least from a 70-mark paper, over 10 mark, or at least up to 14 marks in the combined paper will be maths based, between 14 and 21 marks. It could be even more. In some papers I've seen it go up to 28 marks from 70. But 21 marks, you're already at 30% of the exam. So you've already gone past your grade 4 boundary. In higher, again, you're looking at 30-40 marks, 30-40%. So it's very important that you can do all the maths correctly. Okay, let's have a quick look at this question. So we've got calculate the energy needed to increase the temperature of a 500 gram aluminium block. So first thing we've got to do, we've got an aluminium block which is 500 grams. We're going from grams to kilograms. That means we go from small to big, so we're going to divide by 1,000. So our mass is 0.5 kilograms. And we're going to increase the temperature by 15 degrees. So the change in temperature is 15 degrees, and the specific heat capacity is 900 joules per kilogram per degrees. And we're trying to find out the energy change. So there's our formula, that's our equation, sorry. Mass is 0.5, C is specific heat capacity, that's 900 joules per kilogram per degree. Our change in temperature, now that triangle and theta. So this is the Greek letter theta. The triangle means change in. Theta is the Greek letter for T. So that's temperature. Multiply them all together and you have got 6,750, and really importantly, joules. So I'll let you go through these questions in your own time. There's some exam style questions here. There's another example of a past paper exam style question. Another exam style question. So how can you use apparatus to determine the specific heat capacity of a material? So this is essentially what I've just gone through, but it'll break it down into steps. So number 1, measure the mass. Use the mass pan balance to measure how heavy the material, like the metal block, is. Write down in kilograms. So you've now got the mass. Set up the heater. So place the heater inside the block and connect it to a power pack and jouleometer. The jouleometer will tell you how much energy is being used. So now we know the energy. So don't forget, if you look at the formula, change in energy equals MC change in temperature. So what we're looking for here is change in energy equals mass times specific heat capacity times the change in temperature. So we've now got the mass with step 1. With step 2, we've got the energy change. With step 3, we turn on the power pack. So we add a thermometer. That gives us change in theta, change in temperature. So put the thermometer in. Now we turn on the power pack, wait and record the results. So after 10 minutes, turn off the power pack, note down the final temperature of the block, note down the change in energy, and then you rearrange this formula. So specific heat capacity equals the change in energy divided by mass times the change in temperature. Now I'm going to put that in brackets because it's better to put in brackets to avoid bid mass errors. Because otherwise what can sometimes happen is you get change in energy divided by mass and then it's multiplied by change in temperature. So mass. How do we get mass? We use a balance to measure the mass. How do we get temperature change? We use a thermometer. And you do final temperature, take away start temperature. How do we get energy? Or how do we know how much energy has been supplied? You take a reading from the jouleometer. And then we use this formula to work out specific heat capacity, C. Now, I'm not going to dwell on this question too long, but one thing that you have to be able to do is plot the coordinates of a graph accurately. And then when it says draw a line of best fit, it doesn't have to be a straight line. If you look at the line below, it can also be a curved line of best fit. So here at 60 times 60, which is around there, you got 24.5. So you would put a cross here. And for 120 and 29 degrees, 29 degrees is here, and 120 is around here, so you'd put your cross there. Time 180 is 31 degrees, so that is around here, and 31 degrees is there, so you put your cross here. Then we've got time 240, which is around there. It's quite small, so I might be off slightly. So your final cross goes up here, and then what you're doing is drawing your line of best fit through all of those coordinates. But when you're drawing a curved line of best fit, you do need to go through all the points, other than points which are anomalous results. So if I had a result that put a cross over here, you would put a circle around it and you would ignore that. So power. What is power? The rate at which power is transferred, or the rate at which work is done. So power equals energy divided by time, or power equals work done divided by time, or energy transferred equals power times time. Now, an energy transfer of 1 joule per second is the same as 1 watt, because power is the amount of energy transferred per second. So it's the same as saying joules per second. And another way of saying joules per second is watt. Now, one thing to understand here is work done equals the force over a certain distance. So it's the force exerted over a distance. Now, we've also got here energy. So let's consider potential energy. Potential energy is mass times gravity times the height. Now, what I'm explaining here is something for everyone to understand, but from a higher perspective. It'll still help with foundation, and it'll help with your understanding of knowing the difference between work done and so this W is work done, and this is potential energy. Now, mass times gravity is the same as weight. So I should have done it this way really. Weight equals mass times gravity. So mass times, when we say weight is a force. So that F there is the same as mass times gravity. They're both forces. Now, if I climb up a mountain, and assuming I climb up the mountain, let's say there's a ladder next to it, and I climb up this ladder. When I start at the bottom and reach the top, then the amount I've just moved, could we call that distance? Yeah. But not only is the distance, to get to the top of a mountain, what else can we call that vertical distance? We can call it height. So in that respect, work done and potential energy are the same thing. Now the units for work done are joules, and the units for energy transferred is joules. They are both linked to transfer of energy. The only difference is when you're doing work done, when you calculate work done, you can sometimes get questions that say, why is work done not necessarily exactly the same as what's just been calculated? And that's because due to friction, some of the work done energy, some of the energy is lost. So work done, let's say you got work done of 1200 joules, some of that energy might have dissipated and been lost to the surroundings due to friction. So for that reason, work done as a number is not always the same as potential energy, but you can treat them as interchangeable things as well. So if I said to you the potential energy that I used to climb a mountain was 1200 joules, if I said to you what's the work done to climb up the mountain, you would say the work done is also 1200 joules. So my potential energy equals 1200, my work done also equals 1200. Now I could ask you a follow-up question that says, in reality, why is my work done not 1200 joules? And that's because some of the energy is dissipated, it's lost to the surroundings. Okay, so this is a practice question that now we've got a little bit of time so we can actually have a go at that practice question. Quickly go through it. Now, the electrical generators can provide 1.5 times 10 to the power of 9 watts of power for a maximum of 5 hours. Now, if we just go back for a second, when we say time is measured in seconds. So even in this question, we have to convert the hours into seconds. So how do we convert hours into seconds? First, we times it by 60 to convert it into minutes. How do you convert minutes to seconds? We times by 60 again. And hence we end up with 18,000 seconds. So now what we're trying to work out is power. Now actually in this case we're trying to find the energy transferred. So if power equals energy divided by time, if you want energy to be the subject of the equation, then time, here we've got divided by time. So from an algebraic perspective, we move time to the other side, it becomes multiply. So that means change in energy, or energy equals power times time. So we're just doing 1.5 times 10 to the power 9 times 18,000. Now this is more likely to come as a higher question, but it can come up at the end of a foundation paper as well. So it is beneficial for you to practice using these large numbers. But usually when you've got powers, that only comes in triple. It's very unlikely that questions with powers will come in a foundation paper. Okay, we've got 5 minutes left. We're going to go through this last part, 1.2.1, and then we're going to finish for today. Now, I would appreciate if in the comments you can give me some feedback. If you want to, if you don't want to write anything big, even a rating out of 5 would give us a bit of help. But if you can give us something specific that we can work on or act on, that would be really helpful too. So you've got 4 minutes to consider that. Now, energy transfers in a system. First thing you want to understand here is law of conservation of energy. And the law of conservation of energy is that energy cannot be created or destroyed, only transferred or transformed from one form to another. The total amount of energy in a closed system always stays the same. So what we're saying here is, if I throw a ball upwards and the amount of energy that I throw upwards is with 100 joules of energy. So before we learned that the kinetic energy converts into potential energy. When it does convert into potential energy, it will convert into 100 joules of potential energy. That's within a closed system. Now, if we consider it a system, consider an open system. As that ball is moving upwards, it's going to collide into air particles. And when particles collide into particles, you get friction. So that means some energy will be lost as thermal heat energy. So an example is boiling a kettle. The kettle transfers electrical energy into a thermal energy to warm the water. No energy is lost, it's just changed into a different form. So we've gone from electrical to thermal. So how can we reduce unwanted energy transfers? So I just gave the example of throwing that ball up into the air. If you think of someone who's got a car and an engine, a lot of the time the engine heats up because you've got loads of moving parts. So different parts of the engine will rub against each other. And because they're rubbing against each other, that's friction because particle touching particle. And that energy that is released is lost energy, it's wasted energy. So how can we reduce that wasted energy? What we can do is add lubricants. Lubricants can be applied to the moving parts. They reduce the friction between touching surfaces. That means the surfaces slide past each other with less friction, so less heat energy is lost. That makes the system much more efficient. The other thing we can do is thermal insulation. So common examples are within the home is loft insulation, wall insulation, and insulation around carpets. These methods slow down the transfer of thermal energy, keeping the building warmer for longer. So again, that reduces unwanted energy transfers. Now, next thing to understand is thermal conductivity. So some materials allow heat to pass through them quickly. Now, if something allows heat to go through it quick, then we call that a good thermal conductor. In other words, it's a material that has high thermal conductivity. So if something is a good insulator, then we will say it has low thermal conductivity. So these are different things that affect the rate of cooling of a building. For example, double glazing or triple glazing helps trap in the heat. Wall thickness. So the thicker the wall, the better the insulator. Thicker walls slow down the rate of heat loss. Now in a hot country, a thicker wall also slows down how quickly the heat enters, so it helps keep the house cool. The wall material, the lower the thermal conductivity of the material, in other words, it's an insulator, the better it keeps the heat in. Insulation, so materials like loft insulation help trap warm air inside. And then you've also got draft excluders. They prevent warm air escaping through the gaps around doors and windows. Now, how can we test the effectiveness of materials as insulators. Now, this is the point we're going to stop so we can go through this in a bit more detail tomorrow. I don't want to rush through this, so we're going to stop at this point. I just want to say a really big thank you for everyone that's logged in today, and big thank you to everyone that's given feedback. We will be reading through that feedback and giving you answers and hopefully applying that tomorrow. Now, just a reminder, tomorrow's session will start at 9:45. So if you have any questions, because I think I've answered all the question and answers for today, but if you guys do have any questions, I'll be on from 9:45 and then I can answer those questions live. Okay, thank you very much everyone. Have a lovely day. I'll see you all tomorrow. Okay, good afternoon, everyone. Thank you for joining us in our AMALathon. Welcome to AMALathon 2025. Now, I'm just going to start with a quick introduction as to who we are and what we're doing and why we're doing this, and then we'll make a start with our AMALathon. Now, if you have any questions, just add it to the live chat. If I'm able to answer it as we're going through the work, I'll answer it directly. Otherwise, we have got a specific time for questions and answers. So I'm just going to show you a quick video. It's not going to last long, just to give you a quick intro into who we are, and then I've got a few things to explain and talk to you about, and then we're going to make a start.
Providing you with the equation for success. Activate plus memorize plus apply equals learning. AMALearning, you're about to.
So when with our AMAL method of learning, what we mean by that is initially to activate the brain. And by that, we mean understand everything. So the first step in doing really well in your GCSE exams is understand everything. Now, the way we've provided our notes, and you can get the notes if you join onto our WhatsApp channel, you can get the notes through the WhatsApp channel, and everything is written based on specification points. What that means is when you're doing all of your learning and all of your understanding, if you come across a specification point that doesn't make sense, then we can help you with that. So you can either ask us a question, you can watch one of our animations. Our animations are specific to Edexcel. So if it doesn't make sense, you can watch an animation. You can ask your teacher, you can watch another video. There's lots you can do, but at least that way, you know specifically where the issue is. So today's notes are free. So you can see what they look like and decide if they're right for you. So for those of you that attend the AMALathon live, you'll be able to get the remaining paper one notes for a reasonable discounted price, which is only available during the AMALathon. All you need to do is join our WhatsApp channel, and the link for the WhatsApp channel is in the post, and someone will put it into the chat as well. Now with AMAL, the A stands for, as it showed on the previous slide, the A stands for activate, meaning activate your brain, understand everything. So if you've understood everything, we can move on to the next phase, which is either memorize or apply. So the M stands for memorize, the A stands for apply. What I would advise is initially start going through past paper questions. So once you've understood and through this whole process with the AMALathon, you'll hopefully understand everything a lot better. Then you can move straight onto the apply phase, which is start going through past paper questions. What the past paper questions will do is they will help you to understand what you do and what you don't understand. And alongside that, it will start to give you an understanding of the examiner's mindset, and it'll show you what needs to be memorized, because you'll see common things coming up over and over again. And then the third phase, memorize. Now, I've been using this process. I've been a teacher for over 15 years. I've worked in senior leadership. I've worked in my senior leadership. I specifically worked with improving results. So I worked in schools where results were quite poor and left. And part of my role was then to improve it. And wherever I worked, I did have that positive impact there. I've been head of maths, head of science. Currently, I'm only a head of science. But in the last 15 years, I've always got excellent results at GCSE because I've always used this one process. So with all my students, I always say, and I'm saying the same thing to you guys, understand everything. Activate your brain. If something doesn't make sense, do whatever's necessary to make it make sense. So you've got access to me now for the next 6 days. Keep asking questions. If something doesn't make sense, if you look at this where it says 4:30, there's a question answer session. Ask your questions. I can make more time wherever we need to make more time. So if you don't understand something in as much detail as you would like, you can rewatch the video, you can ask me questions, we'll find a way around it. The second thing is then, I always say to them, is apply it to past paper questions. And when they've started to apply it to past paper questions, what they start to understand is what the examiner wants, and they also start to understand what topics need to be memorized, and the final step, memorize. And I've seen students go from grade 2s and 3s all the way up to grade 9s. So everything we're covering today is not just it's for foundation, so it'll help you get to your grade 5. We're covering higher content, so it'll help you get to your grade 7, 8s and 9s, and we're covering triple content. Now the content for Edexcel is massive. So for biology, we'll be using the next 2 lessons to cover key concepts. Then we'll spend a lesson on B2, a lesson on B3, a lesson on B4, and then a lesson on B5. And on Thursday, we'll have to use the biology session for chemistry, because the chemistry session is quite big, and I do want to spend a full lesson on electrolysis. Now the triple content it kind of interweaves with the combined content. Where I can teach that triple content without any issues, I will cover that triple content. So there's no question answer session today, and there's no triple session today. But tomorrow, after the question answer session, I'll be doing an extra 1 hour for triple content. Now those of you that can attend that 1 extra hour, brilliant. If you can't, I'm still going to go through it, and it'll be recorded at the end. And there's a possibility that topic 5 chemistry and topic 7 physics we might not have enough time to complete it. So with those, I will do my utmost best to complete it within the time. Otherwise, when I do start doing P7 here, I'm going to continue till it finishes. When I start topic 5 chemistry here, I'm going to continue till I finish. Now, with the way I'm going to go through things through the AMALathon is initially I'll read through everything in the slide. The second step, I'll explain things in my own words, and what I will do as I'm going through is I'll ask questions. There's also questions on the slides as well. So my advice to you is think of put in your mind whatever you think is the correct answer, and then compare it to what then comes on the slide and what I'm saying. Just so you can start to see what you do and don't understand. I would start to make a note of all the topics that don't make sense, because it's all linked to specification points. So what we don't want anyone leaving saying is that, oh, I'm not good at key concepts in biology. We want people leaving saying, I understand it all, but I need some more work on 1.3, 1.9, 1.12. No one's going to say, I don't understand cells and control. Instead, we can leave saying, I didn't understand 2.8. I know I've got to work on that a bit more. So it really pinpoints what you do and don't understand. Like I said, the resources for today are free. So as long as if you can join our WhatsApp channel, you'll have all the instructions there. We've decided as a company that we're going to be sending things through WhatsApp as opposed to using emails, because emails can sometimes go into junk. So we don't want to waste anyone's time. We don't want people having to look for these things. Now, if you ask a question, I'll do my best to answer it straight away. Otherwise, we have that 15-minute question answer session every single day. So you've just seen what AMALearning is, what we're about. Hopefully by the end of this, you'll all be able to say AMALearning. You just did. So let's get started. And please ask as many questions as you need. Let's understand this in as much detail as we can. So it's going to start quite basic, because I'm covering every single specification point. And before I forget, I just want to thank everyone that has subscribed, because since yesterday morning, we've gone from 400 and something subscribers to, I think the last count we were just nearly 9,500 subscribers. So I really appreciate everyone that subscribed. I know there's a lot of people that have been emailing to say that they won't be able to join us live, but they'll be watching my recordings. So I really appreciate all the support everyone's given us. We've got a lot of plans in place to help people. We want to make education affordable and easy. As a lot of you will know, tuition is really, really expensive. So we're trying to provide an alternative pathway for that. Now, subcellular, and so let's get started. Subcellular part are the specialized parts within a cell that perform specific roles, specific jobs. These structures are also known as organelles. So from all this part here, the main part is that they're organelles. Subcellular structures are organelles. They can be found both in prokaryotic and eukaryotic cells. So for example, the nucleus, That contains genetic material, which controls cells' activities. The ribosomes, that's where protein synthesis occurs. And mitochondria, that's the site of aerobic respiration. Now, just so you're aware, we are going to continue biology till 2:30. Then we'll have a 5-minute break. We'll do 55 minutes of chemistry, have a 5-minute break, and then we'll do the final hour of physics. Now, we do need to know what each one of these does, because in foundation you'll get asked questions that are specific to to the structures and what their roles are, so their functions. So that's specific to foundation. In foundation, you'll be asked about the functions. You'll be asked to label some diagrams. But from a higher perspective, you'll need to know all of this as a link to some questions. So, for example, ribosomes automatically comes into protein synthesis. Mitochondria and DNA come into a lot of topics. DNA will come into the topic of DNA. It'll come into genetics. Mitochondria will come into the topic of respiration. So having a good understanding of their functions is important. Now, if we very quickly, before we continue, have a look at respiration, we know that mitochondria is an organelle. Okay, now, which two substances have to go into that organelle for respiration? One is glucose, and the other is oxygen. And what does mitochondria do with that? It goes through mitochondria uses glucose and oxygen to do respiration. As part of respiration, it releases water and it releases carbon dioxide. And most importantly, energy. And it's that energy that we use for our life processes. And that is going to come up again and again and again and again. So important that we know that. So, key features of eukaryotic cells. So they have a nucleus. And that means they have a membrane-bound nucleus. So within that cell, they have the DNA enclosed within a membrane-bound nucleus. So all the DNA is inside there. They're larger and more complex than prokaryotic cells, because this is again a very common question. It's that comparison between prokaryotic and eukaryotic. So it is important that you know the difference between the two. They contain a lot more organelles with specific functions compared to a prokaryotic cell as well. They can be unicellular or multicellular. They have membrane-bound organelles. Now, a subcellular structure, for example, the eukaryotic cell is a type of cell that has a nucleus enclosed with a membrane. So this is something that's really, really important now. Because when you're comparing a eukaryotic cell to a prokaryotic cell, you do need to know the difference between the two. And the eukaryotic cell has a nucleus enclosed within a membrane. And it also has membrane-bound organelles, such as mitochondria. The endoplasmic reticulum is an example, but you don't need to know about that. And Golgi apparatus is an example that you don't need to know about. But mitochondria is an example. Chloroplast is a really good example as well. So what's the key learning here? Eukaryotic cell has a nucleus enclosed within a membrane, and it has membrane-bound organelles. For example, animal cells, plant cells, fungal cells, and protist cells. All of these are eukaryotic. Now let's move on to prokaryotic cell. Now, in comparison to eukaryotic cell, a prokaryotic cell is simple. It's a single-celled organism. Now, eukaryotic cells can also be single-celled, unicellular. For example, a protist. But it does not have, and this is the key point here, it does not have a nucleus. And it does not have membrane-bound organelles. So that means it won't have mitochondria. It won't have chloroplasts. Instead, the genetic material is found in the cytoplasm. And it is in a region called nucleoid, but you will not need that for the exam. But it is additional information. Some of our slides do have additional information that will help those particularly moving into A-level biology next year. But for the time being, that's a good bit of extra info, but it's not needed for the exam. So the bacteria is an example. Archaea, which is almost like an ancient version of a bacteria. It's quite a simple, even simpler version of bacteria than bacteria itself. So we need to know the animal cell. We need to know it in detail. So the first thing you need to be able to do is label it. So you need to know that where the cell membrane is this outer layer. There's the mitochondria, ribosomes. The bacteria has ribosomes too, but it's smaller. And we have our nucleus. Now, this is the bacterial cell. This is the flagella. This outer lining is the slime capsule. And then just below that slime capsule, you have the cell wall. So the slime capsule protects bacteria from the environmental threats. Flagella rotate to help the bacterial cell reach another environment. So it's to help it move. Then plasmid is a small circular piece of DNA in bacteria that carries extra genes. And then you've got genetic material, which contains the instructions for growth. We'll learn more about plasmid when we go through genetic engineering. Now it's important that you know the similarities and differences between prokaryotic and eukaryotic cells. Similarities, they have a cell membrane, ribosomes, and cytoplasm. Differences. So you can see the similarities aren't much. Both categories have cell membrane, both categories got ribosomes, both categories have got cytoplasm. But the differences we need to memorize. Prokaryotic cells are smaller. Prokaryotic cells do not have their DNA enclosed in a nucleus. And prokaryotic cells do not have mitochondria or chloroplasts, any organelles which are around the same size. Because mitochondria and chloroplast is roughly the similar size as a bacterial cell. Now, what are the similarities and differences between animal and plant cells? Again, this is a very common question. Both these questions are more likely to come in a foundation paper, but a slightly more difficult version of this could come in a higher paper, so it is important. So both knowing it properly is still important for higher students as well. So similarities, both are eukaryotic cells. Both have a nucleus, a cell membrane, mitochondria, cytoplasm and ribosomes. But plant cells also have a cell wall, vacuoles, and they have chloroplasts. Okay, let's move on to 1.2. So in 1.2, we're looking at specialized cells. Remember, as I explained before, our PowerPoints are based on specification points. So now, if you understood everything I've just gone through, you can, for 1.1, you can give it a tick that you understand it. And 1.2 is linked to specialized cells. What are sperm cells and what are their adaptations for their function? So what helps a sperm cell do its job? And ultimately, what is the job of a sperm cell? The job of the sperm cell is to get to the egg cell and fertilize it. How can it get to the egg cell? It's got a tail, which whips from side to side for movement. Now, what do we have in this region here that helps it move? We've got lots and lots of mitochondria. And if you think back to what we've already said about mitochondria, what happens inside mitochondria, and I'm going to keep repeating this over and over again because it needs to become a process that everyone's really familiar with. Mitochondria, what happens inside? Respiration. And what's respiration? It's when glucose and oxygen are reacted together to release energy, carbon dioxide, water, and energy. And what is that energy used for? That energy is used to make that tail whip from side to side, make that tail move. And it helps the sperm cell move. Once it reaches the egg cell, then we've got acrosomes. They contain enzymes. So if this is the egg cell, when the small sperm cell arrives, it's got a big wall in front of it. So what those, or rather than wall, yeah, we can use wall. Let's use wall as an example. So what those acrosomes are doing, they're cutting down that wall. You can almost see it like a hammer, but the better example would be chemical scissor. It's breaking through so that sperm cell can now enter. And once it's entered, it's going to reach the nucleus of the egg cell. And reaches the nucleus of the egg cell, it's already got half the chromosomes or DNA which are stored here. And then they can combine with the half that's stored within the egg cell. And we now have fertilization, and we've also made a stem cell. That initial cell that has no function other than to multiply. Let's look at the next specialized cell. What are egg cells and what are their adaptations for their function? So they've got a large size and cytoplasm. Now, actually, let's start from haploid nucleus because that's the starting point. Once the sperm cell has fertilized the egg cell, meaning the half chromosomes that came from the sperm cell are combined with the half chromosomes or half DNA that's come from the egg cell. What do we get? We get a stem cell. We get fertilization in a stem cell, which now starts multiplying. After a certain point in multiplying, it starts to specialize. Now, we need energy for for it to multiply, for that growth. And that comes from the large size and cytoplasm. That provides the nutrients for the embryo. So this whole pinkish red region, or peach colored region, is that cytoplasm that provides the energy for providing energy for now this whole cell to start multiplying. So become 2 cells, 4 cells, 8 cells. And it also releases chemical signals. And they release that because that attracts the sperms for fertilization. So the sperm cells automatically swim in the correct direction. What are ciliated epithelial cells and what are the adaptations for their function? Now the cilia beat in a coordinated wave-like motion to move mucus, trap dust, and pathogens out of the lungs towards the throat. The nucleus is usually found towards the lower part of the cell, away from the cilia. So the nucleus is lower down, away from the cilia. It has lots of mitochondria to release energy through respiration. So again, the word mitochondria has come up. So really quickly again, what happens in mitochondria? Respiration. And what's respiration? Is where glucose reacts with oxygen to release energy. Now, what actually happens is this. So let's assume this is someone's trachea or throat, you've got these hairs. Now, as we're breathing air in, air is full of dust. It's full of different bacteria, viruses, different things. What will happen is, as the air goes past the cilia, in one respect, the air gets filtered. So all this dust and all these germs, etc., a lot of it ends up on top of the cilia. Now, mucus is produced. So mucus is the kind of green phlegmy stuff that we sometimes spit out when we're unwell. That phlegm is then flicked upwards. Where does the energy come for the cilia to flick up the phlegm upwards? From the mitochondria. Respiration happens, it provides energy, that gets flicked up until it reaches the top of our throat, at which point we either spit it out, or 99% of the time we actually just swallow it, and our stomach acid then kills off all the germs. Because as this mucus goes upwards and it's getting flicked upwards, all this dust and germs just get attached to the mucus. So what do we need to know about from this point, from epithelial cells? The cilia move. Why do they move? Because of the mitochondria. Very common question is very common to get questions linked to to microscopic technology. So specifically the difference between a light microscope and electron microscope, and also how electron microscopes have helped us to understand cells better. So there's been developments in our microscope technology. Electron microscopes provide greater magnification and higher resolution. This is the bit that needs to be understood. So I can repeat this over and over again, or you can read this over and over again, but you need to know this. This always comes up in exams. Why are electron microscopes better than light microscopes? Why? Greater magnification, higher resolution. Resolution is, I'll give you a quick example. It will come up as a definition afterwards. Magnification, we know, is taking something small and increasing its size. Resolution is this. If I draw two dots 1 centimeter apart, our eyes can look at that and say, yep, I can see two dots. But an electron microscope can see two dots that have a gap of 0.1, 2, 3, 4, 5, 6, 7, 8, 9, this many meters. So that's 1 times 10 to the minus 10, or 0.1 nanometers. So if you had a gap of 0.1 nanometers, now nanometers is 10 to the minus 9, in other words, that number, if you had a gap of that smaller gap between two dots, you could see that gap with an electron microscope. Now that improved magnification and higher resolution means we can see really small structures inside organelles. Now, the next type of question that might come up is the question that, so I want everyone to think about what you think the answer might be while I'm going through it. What are the differences between electron microscopes and light microscopes? So what's the difference between electron microscopes and light microscopes? Electron microscopes, some are positive, some are negative. It's larger, so it's not portable. It's more expensive. We're talking £100,000 plus. But as a positive, it provides a higher magnification. See, the wordings come again. And resolution. So they could ask you a question on why is electron microscope better than light microscope, and that's going to come as part of the answer. They could ask you to compare to each other, and that's going to be part of the answer. So what does that tell us? We just need to memorize it. We need to know it. But you are unable to view live specimens with it. You have to kill the specimen first. Then if you look at light microscopes, they are smaller and portable. That's part of the reason why we have them in schools. Actually, The fact that they're cheaper is one of the bigger reasons as well. They are smaller and portable. That's good. It has lower magnification and resolution, but that's not an issue when we're at school because we don't need to look. We don't need to look at ribosomes. We just need to know how to use the light microscope. It's much easier to use, and we can use it to view live specimens. What are the differences between electron microscopes and light microscopes? So electron microscope is a high resolution imaging device. So this is something we've already covered, so I'm just going to skip through this. Now, and we've already gone through this, why electron microscopes more advantageous or better for viewing cells. So they've got higher resolution, and that's up to 0.1 nanometers, and they provide greater magnification, up to 2 million times. Now, light microscopes usually max out at 1,500 times. As in, you can magnify something 1,500 times, but with the electron microscopes, it's 2 million times better. So here's our key terms, and I've already defined these, but let's quickly read through them. Resolution is the ability to distinguish between two separate points in an image. So if you draw two dots, if the gap between the dots was 0.1 nanometer, you would see that gap with an electron microscope. And magnification, the ability to enlarge an image compared to the actual object. So if something is this big, when you magnify it, it makes it bigger so you can see it clearer. Now, alongside this, you need to know how to calculate magnification. Now, magnification equals size of image divided by size of the real object. If we rearrange this, size of the image equals size of the real object multiplied by magnification. That's the one that I would advise that you learn. The reason being, if we say size of image is I, and if we say, let's rub this out so I've got space, size of the real object is the actual size, or A, and magnification is M, you can just remember it as I am. That's much easier to remember, I am. So if you're in the middle of an exam, it makes your life a lot easier. Now, the top one here, magnification, image size, actual size. So if you're working out magnification, you can remember it as Mia or Maya. I'm sure everyone knows at least one Mia or Maya. And if you're trying to work out size of image, you can remember it as I am. And if you're trying to remember it as the actual, because actual will equal image times magnification, so aim. Let's take that and have a go at an actual past paper question. So it modified slightly, but it is based on a past paper question. A bacterial cell is 0.04 millimeters long. So out of I am, which one do we have from I am? It's 0.04 millimeters long. So that is the actual. So we know what the actual is. And it has calculated the length of the image. So image we're calculating, so that's question mark. And it says the cell has been magnified 500 times. So we know this one as well. So we've got two bits of information and we're trying to find the third bit. That's the magic triangle, if anyone likes to use magic triangles. So first thing, we've identified what units we already have. We know the actual size is 0.04. We know the magnification is times 500. Let's now substitute the numbers in and calculate. So A is 0.04. Magnification is times 500. Multiply them together, we got 20, and don't forget the units. usually that's a mark. If they haven't already given the units, it's a mark. So make sure you put the units down. 1.4 and 1.5. Milli. So if we use that as an example as meters, millimeters, it's just that small m. That stands for milli. That symbol there stands for micro. So almost like an upside down H or wrong way around Y. N is for nano. P is for pico. Milli is 10 to the minus 3. So for example, 0.001, or dividing something by 1,000. Micro is 10 to the minus 6, or dividing something by a million. And nano is 10 to the minus 9, or dividing something by 100 million. Or we've got pico, 10 to the minus 12, which is the same as 1,000 million. What is important is that you know how to convert from one unit to the next. So this is a really good way of learning this. When you're going from something bigger, as in you're going this way, you're going from a bigger unit to a smaller unit, then you multiply by 1,000. If you're going in the other direction, so for example pico to nano, you're going from small to big, then you divide by 1,000. So let me repeat that. When you go from, for example, millimeter to micrometer, you go from a bigger unit to a smaller unit. So what do? We multiply by 1,000. If you're going from a smaller unit to a larger unit, so micro to millimeter, then what do? We divide by 1,000. Now, it's important that you know this, because the difference in knowing this and not knowing this is multiple marks. It comes up a lot, particularly in higher. When I say multiple, in each paper, there's going to be at least 2 or 3 marks worth of conversions. So you're looking at 6 marks across both biology papers. They're not marks that are worth losing. You need to know these marks. So if we go from milli to micro, we're going from bigger to smaller. Micro to nano, we're going bigger to smaller. Nano to pico, bigger to smaller. What do? We multiply by 1,000 each time. And when we're going in reverse, we are dividing by 1,000 each time. Memorize this slide. If you don't know it, memorize it. So let's have a quick look at this question. The width of this root nodule is, or a width of our root nodule is 7.5 millimeters. Give the width in micrometers. Now, in this situation, we're going from 7.5 millimeters, but we are changing to micrometers. We're going from bigger to smaller. When we go large to smaller, what do? We times by 1,000. So then the answer is going to be 7,500 micrometers. .6 core practical. Investigate biological specimens using microscopes, including magnification calculations and labeled scientific drawings from observations. Now with core practicals, the best thing to do is on YouTube, if you Google the name of the core practical, or if you just Google this investigate biological specimens using microscopes, and write Edexcel, you'll get an animation, not an animation, so you'll get a video, and that video has been specifically made by Pearson and by the exam board. That's not something I can go through in as much detail, because during this, what I'm going through is only the theory. So to actually physically see any of the core practicals, it's best to use that website. sorry, the YouTube channel made by Pearsons and Edexcel, but I'll still go through the theory. But if there's anything that's not understood or you want to understand better, you just need to ask. So, how do you prepare a slide of cells to observe under a microscope? This usually comes up as a 3-mark question, and it's actually quite a common question in both foundation and higher. So again, I would advise that you memorize this. You take the onion skin, peel thin layer, or you can take cheek cells and use a cotton swab, or you could use pond water, where you can use a dropper like this to get the water. Whatever you've taken, we call that a specimen, and we put at the center of a clean slide. We use filter paper, and then what we do is we add our iodine solution, add a couple of drops, and that stains it. Then you put a cover slip on top. When you put the cover slip on top, sometimes what happens is you get a bit of excess liquid, excess starch solution, or whatever stain that you used. Now, what you do there is you take a small amount of tissue paper and it just absorbs that excess liquid. Otherwise, it can ruin what you're looking at. And alongside that, you need to know how to use an actual microscope. And this is again a common question. How would you set up and use a microscope to observe cells? So you place the microscope on a flat surface, plug it in, especially if you're using a light microscope. Now that might seem like a trivial point, but I've seen it in mark schemes. It is a core part of using it. If you don't plug it in, it doesn't work. You've got some that have got mirrors. So with those ones, you would just point the mirror in the direction of the sun. That would get you the mark 2. Eyepiece and adjust the coarse focus. So you've got the eyepiece lens that you're looking through, which usually has a magnification of ×10. And you adjust the coarse focus. Now that coarse focus causes it to move up and down. Now initially you would take the objective lens so it's touching the stage. Not touching, but as close to the stage as possible. Without it touching the slide that you just made based on the previous slide. Then you start turning the coarse focus so it is really, really close. Then you slowly bring it up till you can kind of see the image. The fine focus brings up the objective pieces more slowly than the coarse focus, and it helps you get that really clear image. And then you would draw whatever you can see. So if you can see multiple cells, you're going to draw them all to the best of your ability. And then you would do the same thing. Initially you do it at ×10, then you try it at ×40, ×100, depending on how good the microscope is. But again, you need to know those steps. Now there are some risks. And again, if you've got a core practical exam question, you may get questions within the evaluation, or you may get questions linked to hazards. It's a very common question, so you need to make sure you know that iodine solution, if you use that as your stain, it can be toxic, it can irritate the skin, the eyes, and it can stain clothes. So we only use small amounts. We can wear gloves, or if you're using a dropper anyway, it is not massively a problem if you don't have gloves on. And if it spills, you clean it immediately. If it touches your skin, you wash it off immediately. Then, if, for example, using pond water, you can get contamination from bacteria. And it's important then to wash hands after handling. Dispose of samples properly. Now, describe how this is an actual past paper question. I think it was a 4-mark question. Describe how this equipment could be used. Also, just before I continue, this is the kind of question that you would find at the end of a foundation paper and at the beginning of a higher paper. So describe how this equipment could be used to prepare a slide of onion cells to view under a microscope. So again, this is the kind of answer, whenever you're doing past paper questions, if you come across an answer like this, it is always beneficial to just memorize it. Because look, it's giving you every single step. Pretty much almost the same as what we just went through. Add a drop of water to the microscope slide. Okay, we know that from what we just learned. Place a thin layer of tissue on the slide. The tissue comes from here, from the onions. Stain the tissue with a couple of drops of iodine solution. So again, we've just covered that. And then finally, place the cover slip on top. And we've got our 4 marks done. Describe how this equipment, so if you now compare that to the mark scheme, because that's what we've already learned, you know, that's 4 marks out of 4, or 3 marks out of 3. What we didn't cover in what we were going through stuff, we didn't cover the forceps, because you can use forceps to pick up the thin layer of onion cells. But we've covered placing onion cells on the microscope slide, adding a drop of stain, placing a cover slip on top, and lowering the cover slip down slowly at an angle. Now, the student squashed the root tip on the microscope slide to spread out the cells. The slide was placed on the stage of a microscope. Describe how to use the microscope to obtain a clear image of the cells. So this question is just saying how to use a microscope. Again, it's just worth memorizing these kind of questions. Place the slide on the stage and use the lowest power objective lens. Turn the coarse focus wheel to bring the image into focus. Increase the power of the objective lens to increase magnification. And done. That's your 3 marks. So remember what we said before? Well, this case says 2 marks. Saying that switching the lamp on, you get a mark. Starting with the lowest objective lens, then using the fine focusing wheel or the coarse focusing wheel to obtain a clear image. Any of those 3 get you the marks. And it's only a 2 mark question. Okay, we're moving now on to from 1.7 to 1.11. The aim of today's session is to at least get up to 1.12. 1.12 is actually a triple topic. So if we do have time, we'll add that triple topic in. Otherwise, we'll be covering that triple topic tomorrow. And that will be covered during the triple session after we finish the physics session. So like I said to you guys before, it's 1:30 to 3:30. 1:30 till 4:30. Then from 4:30 to 4:45, we're doing question and answer session. And then from 4:45, there's an hour session just for triple. And if you can't make the triple session, it's not a problem because it'll still be recorded and you can watch in your own time. I appreciate for some people sitting for 4 hours might be a long time. The other thing triple students can do is if there's a topic that's quite straightforward, they can move away from the screen and then come back for that triple content, because a lot of the time triple students are really good with the combined stuff and sometimes not so good with the triple because it doesn't get covered as much sometimes. So let's start with the mechanism of enzyme action using the active site and enzyme specificity. So how do enzymes work? So it is actually a biological catalyst, and we're going to learn more about catalysts when we get to the topic in chemistry, rates of reaction specifically. It speeds up the reaction without becoming part of the reaction. So how do they work? Each enzyme is specific to its substrate. So here we go, we've got an enzyme on the left side, substrate on the right side. You can see how they fit into each other. That substrate fits into the active site. Now this part of the enzyme is called an active site. And that's where weak chemical bonds are made between the enzyme and the substrate. And when that substrate attaches into that enzyme, it makes an enzyme substrate complex. At which point the substrate can be broken down or different substrates can be joined together. Now an enzyme is specific to its substrate, means it only works with one substrate. For example, in digestion, respiration and DNA replication, one enzyme will only work with one substrate. So a protease enzyme, it breaks down proteins into amino acids. It only works with proteins. Protease will not work with carbohydrate, it will not work with lipids and fats. So there's the enzyme, there's the active site. Once they've joined together, we now have, so this is your first phase. They join together, and now we have an enzyme substrate complex. And that makes it easier to break down substrate, and we have the products. And we have the enzyme at the end, unchanged. And that's the whole point. Enzyme is a catalyst. Catalysts they increase the rate of reaction, but they don't become part of the reaction. And that's what the enzyme is doing, but on a biological level. And now that enzyme is free to make an enzyme substrate complex with another substrate. We actually call this a lock and key model. The reason we call it lock and key model is just how, in the same way, your key can open a lock, and your key is specific to the lock to your front door. The substrate is specific to the enzyme. Now explain the reason. Like I said to you guys before, whenever a question comes up, think about it. Explain the reason why no product will be formed if the enzyme is mixed with substrate B. So this is an actual past paper question, or linked to a past paper question, where there was an image of substrate A and there was an image of substrate B. And the question said, why is it that substrate B wouldn't react but substrate A would? And this goes back to the fact that substrate A has a specific shape which fits into the active site of the enzyme. So an enzyme substrate complex can be made. The substrate B does not fit into that active site, so an enzyme substrate complex cannot be made. And this is the actual mark scheme. So if you look at it, the active site of the enzyme, that gets you one mark, will not fit substrate B. That means the enzyme cannot cause the reaction to occur. So look, the question might seem complicated, but if you look at the answer, it's straightforward. That why is substrate B not reacting with the enzyme? It's really simple. Substrate B does not fit into the enzyme's active site. If it doesn't fit, no reaction. Same way if you had your door key and you take it to school and you try and open one of the teacher's doors, it's not going to work because your home's door key is not specific to the lock that opens the door of one of your teacher's classrooms. Explain how enzymes can be denatured due to changes in the shape of the active site. So when we say a denatured enzyme, what we actually mean is it's an enzyme that has lost its shape, making it non-functional. So the enzyme cannot regain its shape. So here we've got an active site that fits the substrate. But if it denatures, it means the shape of the active site changes. Now that substrate is not going to fit. Now, any exam questions linked to denatured enzymes will generally require you to write three things. It's always three things. When the enzyme denatures, and I would advise you memorize this, because whenever the word denature comes as part of your answer, always write what I'm about to tell you to write. If you add these three things to it, you're going to get the marks. A lot of people, they just write, oh, the enzyme denatured, so it stopped working. You should always add these extra bits too. When the enzyme denatures, so the first thing is the active site changes shape. Now, if the active site has changed shape, the enzyme no longer works. So the wording of this has changed slightly, so I'm just going to explain this in my own words. So when the enzyme denatures, the active site changes shape. So first things first, the active site has changed shape. That means the substrate can no longer fit. That's the second thing that you should say. So the active site has changed shape. The substrate no longer fits. This is a substrate here. If the substrate no longer fits, then no reaction. Now, denaturing of enzymes can happen due to high temperature or extreme pHs, and that causes the enzyme's active site to change. So boiling the enzyme stops the protein from being digested. So enzyme, if you boil it, so if you take it to a really high temperature, so explain how boiling the enzyme will stop the protein from being digested. If you boil it, you will change the shape of the active site. If you change the shape of the active site, the substrate no longer fits. If it no longer fits, you no longer have a reaction. So the enzyme is denatured, the active site changes shape. That means the substrate will not fit into the active site, which means there's no reaction. 1.9. So now we're looking at the effect of temperature, substrate concentration, and pH on enzyme activity. So describe and explain the link between the impact of increasing temperature, pH or substrate concentration on enzyme activity. So first thing we're looking at here is enzyme activity temperature, and we'll look at pH and we'll look at substrate. So let's look at these one at a time. Now you're going to get two different types of questions linked to this. The first one is a describe question. So if you get asked to describe this or this, really simply put, and I'm going to explain this in a bit more detail, what's actually happening is this. As the temperature increases, enzyme activity increases. Then we reach optimum enzyme activity. Then as the temperature continues to increase, enzyme activity decreases. At no point have I explained why. All I've done is describe what's in front of me. So if the question says describe, then this is the description. As the temperature increases, the rate of enzyme activity increases. When does it stop increasing? When it reaches optimum temperature. As the temperature continues to increase, what happens to the rate of enzyme activity? It decreases, it falls. Now, if I'm explaining the science behind the same thing, it becomes a little bit different. So in an explanation, you still put the description. So note how I've still got a description there. As the temperature increases, the rate of enzyme activity increases. But now we're going to explain that description. We're not going to just say the description, we're going to explain it. That's because when you increase the temperature, you're giving energy to the particles. So the particles got more kinetic energy. So thermal energy converts to kinetic energy. If they're moving faster, you got more collisions. More collisions, more reactions. And that happens until we get to optimum temperature. But now, look, we're describing again, as the temperature continues to increase, the rate of enzyme activity decreases. Why? So look, with an explanation, we're saying the description. That's the description. The two bits I've circled are descriptions. And this bit's a description as well. But now we're explaining the science. The high temperature has caused a change in the shape of the active site. Now, if it's changed the shape, in other words, what's it done? It's caused the enzyme to denature. If it's caused the enzyme to denature, it's changed the shape of the active site. So AS, active site. If it's changed the shape of the active site, the substrate no longer fits. If it no longer fits, you have no reaction. And that's what's on this slide here. This is a slide that should have been there before. Due to high temperatures or extreme pH, the enzyme's active site changes shape. Because the substrate no longer matches the active site, an enzyme substrate complex cannot form. Enzyme no longer works, the reaction slows down or completely stops. Whenever you write about denature, you automatically write this as well. They have to go hand in hand. Okay, now I know I said we were going to finish at past and then have a 5-minute break. We are going to take the 5-minute break as soon as topic 1.9 is finished. So again, let's describe this. Description is going to be very similar. As the pH increases, the rate of enzyme activity increases until we reach optimum pH. As the pH continues to increase, the rate of enzyme activity increases. So look what we've got here. We've got 3 bits of description. That's all you need. No explanation needed at all. It's not asking for it. And if we now explain, here we've got a description. Here we have a description. And here we have a description. This is the only part that needs an explanation. When you increase the pH has caused a change in the shape of the active site. In other words, what has the pH done to the enzyme? It has caused it to denature. And if it's caused it to denature, we always need to write down three things. The enzyme is denatured because of a high pH, which means its active site has changed shape. That means the enzyme active site has changed shape, then the substrate will no longer fit. If it no longer fits, there is no longer a reaction, so the rate of reaction decreases. And finally, we've got substrates. Now, what it shows here is when a substrate concentration, when the substrate's concentration, now concentration means how much of it you have. So here you only have one substrate, so that's a low concentration. If I had 100 of them, then there'll be a higher concentration. If I had 1,000 of them, that's an even higher concentration. So when substrate concentration is low, lots of enzymes have empty active sites and the rate of reaction is slow. Remember, once this substrate attaches itself, that enzyme now is in use. Once it's made the product and that substrate is no longer taking up the active site, the next substrate can fit into that gap. Now, when substrate concentration is high, most enzymes have active sites containing substrates, and so the reaction can't go any faster. So we have a maximum to this reaction. Why do we have a maximum? Because all the enzymes, when you have a high concentration, each one of them is full with a substrate. If each one is full with a substrate, then all the additional substrates, you can keep increasing it as much as you want. There's no space for this new substrate. If we now increase the concentration further, there's no space for that substrate until one of these enzymes is broken down into products and it frees up space. So initially, when you've got a low concentration, substrate concentration, there's lots of empty enzymes. So as you increase the concentration, the enzyme activity also increases. When we get to a certain amount of substrate concentration, the amount of enzymes becomes like a limiting factor. So you can't increase the enzyme activity beyond that point. Okay, tomorrow we are going to continue from 1.10 in biology. So thank you very much everyone for the time you've just given, how much you've concentrated and learned. So really appreciate it. We're going to restart in 5 minutes. That means we're going to restart at 2:39. So I see you all at, sorry, let's just call it 2:40. So we're going to restart at 2:40. By the way, because of the way we're running this, we are sometimes going to be 5 or 10 minutes off schedule. So I hope that's okay for people. But I do understand people will just log off if they need to log off at a certain time. So like I said, I'll see you all we'll have a break till 2:40, and then we'll move on to chemistry. Thank you very much everyone. See you in 5 minutes. Okay, welcome back everyone. So what we're going to do now is we're going to continue with chemistry, and we are starting with topic 1. So we're going to be trying to cover all of the key concepts in chemistry across 3 days, because key concepts in chemistry comes in both paper 1 and it comes into paper 2 as well. So it's important that we've properly understood. So like I explained before, there's no question and answer session today. And tomorrow I'll start the triple content coverage as well, hopefully. Now, today we're going to finish as much of key concepts as possible. Then we've still got 2 more days to go through. Well, on the 3rd session, that's where we'll mainly be focusing on the maths side of things. And in this 2nd session, we'll mainly be focusing on ionic bonding, covalent bonding and metallic bonding. But keep asking questions. There was a question which I just saw, which I'm going to really quickly answer as soon as I get back onto this. We'll mainly be focusing on. And the question was, this is from biology, so will we be given the graphs of enzyme activity in the question? And the question is yes. So if the question comes up, you'll get the graph in front of you and it might say describe what you can see, or it might say explain. But what I was trying to get you to understand is the difference between the describe and the explain. And it is nice to see that some of my students are on. Excellent. Right. Let's get started. Please keep asking questions if it doesn't make sense. And we're going to be on chemistry now till 3:40. So we're going to give it a full hour. So how has the atomic model changed over time? Now this will come up again in physics, and but when we do it through in physics, we'll go through it in not as much detail. So the model of the atom has changed over time. So initially, if you look at Aristotle, he was an old, well, don't think the word old is needed. He was a Greek thinker. So we're talking thousands BC. He said everything was made of 4 things: earth, air, fire, and water. Other Greeks went beyond that, so it wasn't just Aristotle who had a theory. Other Greeks thought if you took, for example, a piece of paper, ripped it in half, 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 and you kept ripping it in half, eventually you'd end up with what they call atomos, something that can't be divided any further. Now, Dalton took that idea and went one step further. He started to explain that in a bit more detail. He suggested atoms were tiny spheres that could not be divided. So they were atomos, they were indivisible, sorry. Now they didn't know about electrons at that point. Then came J.J. Thomson. He discovered the electron. So he discovered that there was a negative element to everything. And he suggested the plum pudding model. This model said that the atom is a ball of positive charge with tiny negative electrons stuck inside it. So he was saying this bit I'm shading blue was negative. So all this bit's negative, and the bit on the outside was positive. Now, I suppose in the modern day and age, plum pudding is not something that we're familiar with. So you could look at it as a cookie, a chocolate chip cookie. The cookie part is positive, and the chocolate chips are the negative part. Now, we can't rename this as the chocolate chip cookie model, but that's an easier way of understanding it. Then came Rutherford's experimentation. So just excuse me 1 second. Then came Rutherford's alpha particle scattering experiment. Now, what we learned from this, right, apologies, it's ended up going straight to the back, right, back to the start. Right, so Ernest Rutherford, what he did was he found alpha particles are gold atoms. Now what he did with the gold is he made it as thin as possible. Now the thing with gold is that if you got a gram of gold and stretched it from both sides, you could probably stretch it for a mile, as you could make a mile's worth of gold wire from gold. It is very ductile. So you can, when you hit it with a hammer, it's very malleable as well. You can really thin it out, so it's only a few atoms thick. Now the assumption would have been if they find alpha particles at it, or they'll all bounce off. Now an alpha particle is a particle that's made of 2 protons, so that's positively charged, and 2 neutrons, which are neutral. Now the assumption was when they try this tiny particle at it, everything will go straight through. No, it will bounce back, sorry. What they got instead was majority of them went straight through. Now the fact that majority of them went straight through, that showed them that most of the atom must be an empty space. So they started to assume that the atom is like that, and majority is now empty. They noticed some of them bounced right back. The fact that they bounced back showed them that there must be something in the center that's really tiny and heavy. Now, the reason they thought that was because if it bounced back, there must be something that is bouncing off. The second thing, only one bounced back out of 18,000. So if only one's bouncing back from 18,000, that means the majority of it is empty space and you've got a tiny thing in the center. So I'll just draw it here again. So it had a tiny thing in the center and they called that a nucleus. The next thing they noticed was that when they fired these particles, some of them got deflected. Meaning when they went towards the bit that was bouncing back, they were repelled in a different direction. Now alpha particles are positive. If you get 2 magnets, the positive side of this magnet and the positive side of this magnet, what will they do? They repel each other. So then if the alpha particle is positive, they therefore assumed that whatever this nucleus is in the middle, it must also be positive. Now at that time, they just assumed the negativity was like almost like a cloud of negativity around the outside. So using this alpha particle scattering experiment, we went from the plum pudding model, or what we called the chocolate chip cookie model, which please don't write in the exam, and we went to this new model which they called the nuclear model. And the nuclear model, the conclusion was there's a tiny nucleus that was positive and the majority of the atom was empty space, and we called it the nuclear model. Here. Then after Rutherford came Niels Bohr. He improved it because his experimentation showed that there were electron shells and that the electrons were within those electron shells are fixed orbits. And then after him came James Chadwick, who discovered and proved that neutrons exist. So in the nucleus, he took away that it was just positive and he added neutrons. As it wasn't just protons, he added the neutrons in there as well. Now, the kind of questions you're going to get on this topic is you might get a question that'll say, what's the difference between the plum pudding model and Rutherford's nuclear model? You might get, what's the difference between the nuclear model and the most up-to-date model? And the difference is we've now got electron shells and neutrons. So it's always a comparative question. So this is a really good example here. Give two differences between Dalton's model. So remember, let's quickly go back. What was Dalton's model? This one. Dalton's model of atoms and today's model of atoms. Now let's say you've forgotten Dalton's model. Not a problem. Just write down what we know about today's model of atoms. We know that what have we got in the center? We've got a nucleus, which is a small part of the atom. We've got lots of empty space. We've got electron shells with electrons inside them. The nucleus has got positive protons, and it's got neutrons. Around it in the electron shells, we've got electrons. So we've got all this stuff that we can say. So John Dalton lived over 200 years ago. John Dalton suggested early model atoms. When Dalton first described atoms, he said that all elements are made of atoms. So he was correct about that. Atoms are not formed from any similar particles. All atoms of the same element are identical. Now, the main thing is what we just discussed before that. Atoms are made of subatomic particles, meaning they're made of protons, electrons, neutrons. Atoms have a nucleus. Atoms contain protons, neutrons, electrons. Atoms of the same element can have different numbers of neutrons. So just quick question here, do we need to know all of the scientists for the particle models? No, not really, no. Occasionally I've seen the odd question which says, and I've only seen it once, where it said who discovered a neutron, or what did Niels Bohr discover, some along those lines, but you don't need to know the names of the scientists. You do need to know how the models have changed and what the difference between the models is. Now, you could have spoke about Dalton's model talking about the atoms are indivisible, as in they can't be divided any further. Now this has no link to the plum pudding model, so you don't need to mention it, because we're only talking about Dalton. If it said J.J. Thomson, then we're talking about the plum pudding model. But it won't say J.J. Thomson's model, it will say plum pudding model. Okay, structure of the atom. What is the structure of an atom? An atom has a tiny center called the nucleus, which holds protons and neutrons. Electrons move around the nucleus in energy levels called shells. So this is what we know. And then you've got subatomic particles. The relative mass of a proton is 1. The relative mass of a neutron is 1. Now the electron, if you compare it to the proton, it is 1,836 times smaller in mass. So the best for, it says here very small, almost 0. But if you get this as a question in an exam, and this is more likely to come in foundation, you write 1 over 1,836. Now, why are atoms neutral? Because protons have a charge of +1, electrons have a charge of -1. Now let's really quickly look at carbon as an example. So we've got carbon 12. Now, that bottom number tells us the number of protons. The top number is protons and neutrons. So how many protons does carbon have? Protons, it has 6. Now, in an atom, the number of protons and electrons is always the same. So in terms of electrons, it has 6. And how do you work out the number of neutrons? You do the top number, which is the mass number, take away the atomic number, or proton number, which is the bottom number. So top number is mass number, which is protons and neutrons, and the bottom number is proton or atomic number, which is P, which tells you the number of protons. So 12 take away 6 is also 6. Now the number of neutrons is not always the same. It is in this case, but not always the same. We'll do some examples when we talk about isotopes where you'll see different number of neutrons to protons. Now, that means we've got 6 protons. So we've got 6 plus 1s, which is 6 plus. We've got 6 electrons, which means we've got 6 minuses. If you've got 6 pluses and 6 minuses, what's your overall charge? 0. And that's why atoms are neutral. Now, this has got nothing to do with neutrality, so we can ignore that title. It's just talking about the size of atoms. Now, the nucleus there, compared to the overall size of the atom, the nucleus is usually 1/10,000th, as it's 10,000 times smaller. That's as much as you need to know. There have been questions linked to this, but they've given you both numbers. So there's a question that said the atom is this big, 1 times 10 to the minus, the radius of the nucleus is 1 times 10 to the minus 14, but the radius of the atom is 1 times 10 to the minus 10. How much bigger is the atom? And then you're just dividing both numbers. And you're just doing 1 times 10 to the power of 14 divided by 1 times 10 to the minus 10. No, sorry, wrong way around. So minus 10 at the top, minus 14 at the bottom. And that equals 10,000. Meaning that this is 10,000 times bigger than this. So where is most of the mass concentrated in the atom? So it is concentrated in the nucleus. So this is really simple, this one. What's in the middle? Protons and neutrons. And the relative mass of a proton is 1. The relative mass of a neutron is 1. The electrons are on the outside. They only have a relative mass of 1 over 1836. 1836. So that part is not going to be a big part of the mass. So we know also from Rutherford's experiments, that's where the mass of the atom actually is. That's what we need to know. Mass number. So mass number is, let me give you an example. Let's put carbon 12 there. That's the mass number. It's the number of protons plus neutrons. So this is the symbol, carbon. You use a symbol as 1 or 2 letters, but the first letter is always capital, and the second letter is smaller. Now, in chemistry, our aim today is to get to 1.20. And like I said, mass number is the number of protons and neutrons within the nucleus of an atom. Atomic number is the bottom number. So again, let's use carbon as an example. I know we've already said this multiple times now. This is the atomic number, the number of protons, and this top number is the mass number. Isotopes. Now, before I even put anything up on isotopes, I'm going to explain this in my own words, and then I'll put up the details. So here we've got carbon 12 and carbon 14. Now let's look at carbon 12 first. Protons is the bottom number, so that's 6. Electrons is also same, 6. Remember, because the atom's neutral. So there's just a quick question here. Is the relative mass of electrons negligible? Yes, that's an acceptable answer in a mark scheme. You can write negligible or tiny or really small. However, I would advise that you always put electrons as 1836. Because in some mark schemes I've seen it where they've not accepted tiny So I would always stick to that number. But in some mark schemes, it's tiny, negligible, etc. is allowed. Yeah, once this recording's finished, once our session's finished, it will go onto YouTube. So you will be able to watch this again and revise from it. And the number of neutrons is top number take away bottom number. So neutrons also equals 6. Now if you look at carbon 14, again, protons equal 6. Now here really we don't need to worry about electrons. I'm just going to cross that out. And neutrons is top number take away bottom number. 14 take away 6 is 8. Now they're both carbon. And why are they both carbon? Because they both have the same number of protons. It's the bottom number that tells us what element it is. So they have the same number of protons, but what do they have different? They have a different number of neutrons. That's what an isotope is. So it's the same atom, the same element. They're both, so not the same atom, they're both the same element because they have the same number of protons, but they have a different number, a different mass number or a different number of neutrons. So now if I erase all this and I show you the definition, if you want to quickly read the definition while I rub this out, you'll see it's exactly what I've just explained. So atoms that have the same number of protons, so same atomic number, but a different number of neutrons, so a different mass number. Carbon 12 and carbon 14 are isotopes because they both have 6 protons, but they have a different number of neutrons. So if you get an exam, so this is an exam question. Explain the meaning of the term isotope. And it's exactly what we've just covered. This comes up, I think I've seen this nearly every year. In one form or another, it gets asked over and over again in lots of different ways. And the answer is, same number of protons, different number of neutrons. Couldn't be simpler. Mass and atomic number. So what does each tile on the periodic table tell us about the atom? Now this is something which I've just explained to you how to do, but let's look at this again. Let's look at sodium. So here we've got Na. At the top, the mass number is 23, and the bottom number, the atomic number, is 11. Now you need to be able to break that down into number of protons, number of electrons, and number of neutrons. Now when I'm going through stuff, if you can have a go yourself, if anyone's really fast at typing, they can even put it into live chat and I'll be able to see it. But if you can't, it's not a problem. At least think of what the answer is. So this bottom number, 11, it always gives us the number of protons and the number of electrons. Remember, our atoms are neutral. And how do you work out neutrons? Top number, take away bottom number. What's 23 take away 11? 12. So that's what we're showing here. Total number, so number of protons is 11. Number of neutrons is 12. So let's have a look at magnesium. So magnesium on your periodic table will look like this. So let's look at one at a time. Protons is that bottom number, so it's 12. Electrons is always the same number, so again it's 12. And neutrons is the top number take away the bottom number. So what's 24 take away 12? It is 12. And the information there just explains what I've just explained. So if you've asked for the notes, I'm assuming I mentioned this at the beginning, but if anyone's looking to get hold of the notes, all you've got to do is join our WhatsApp channel. Now the beauty of WhatsApp channel is we don't get your number. So we won't know what your number is, you've just joined the channel, and that way we can transfer the notes to you on the channel itself. For those that want to buy the resources, there'll be instructions on the channel itself. So there's someone dealing with that right now. So you can do it now, you can do it at the end of the lesson, any point. there'll be someone that has access to the WhatsApp channel, and they'll contact you and help you with that. Isotopes and mass numbers. So again, I'm going to explain this in my own words rather than using what's on the slide, and then I'll show you what's on the slide after, because it's easier to understand that way. Now, let's say you've got chlorine. In nature, you've got two types of chlorine. Chlorine 35, so it's a mass number of 35, and we also have chlorine 37. But on the periodic table, chlorine looks like this: 35.5. And that's because it's an average of chlorine 35 and chlorine 37. And that's because we have 75% of chlorine 35 and 25% of chlorine is chlorine 37. Now if we do the calculations for this, so what we're going to do is 75%, so 75 divided by 100 times 35, so 75%, and we multiply that by 35, and to that what we're going to add plus 25% of 37, so that's 25 over 100. So when you want to look at the percentage of something, you divide it by 100. Percent means out of 100, times that by 37. And what does that equal? It equals 35.5. And that's why some mass numbers are not whole numbers. So again, I'm going to rub this out so you can read what's there. And what's there is just an explanation of what I've just shown you. Chlorine has different types of atoms, isotopes, which is why its atomic mass is not a whole number. Chlorine is made up of two isotopes, chlorine 35 and chlorine 37. Together they give an average atomic mass of 35.5. So it's all about an average. Calculating the relative atomic mass. So why do some elements have atomic masses that are not whole numbers? Now this is what I've just explained, and it's just a calculation that shows exactly the same thing. The only thing that they've done differently is where I put 70 over 100, where I would have put 70 over 100, they've already done that calculation, then put it at 0.7. So if you get hold of the notes, or when you get the notes, so if anyone's even watching a recording of this, you can contact, if you look at our posts, it tells you how to join the WhatsApp channel. A lot of what we'll be doing is through the WhatsApp channel, simply because with emails there's a lot of issues. Sometimes it goes to junk mail, etc. So it's good if you can join the WhatsApp channel, and we can communicate through the WhatsApp channel that way. You'll be communicating with someone from the AMAL team, and that way you can have got this in your own time as well. Okay, the periodic table. So how did the periodic table develop over time? So now what we're looking at is Mendeleev, and how things have gone from Mendeleev's time. Well, this is actually before Mendeleev. Then we got to Mendeleev, and how we've then moved on to the modern periodic table. So the first version of the periodic table looked like this, where people were trying to put things that reacted similar to each other in groups. So lithium, sodium and potassium are all group 1. They react in a similar way. Fluorine and chlorine react in a similar way. Beryllium, magnesium and calcium react in a similar way. So there was some logic behind it, but it just wasn't quite fitting. And it was incomplete. Some elements were placed incorrectly because they had not discovered a lot of the elements. In fact, a lot of elements weren't discovered until electrolysis improved. or until they discovered what electrolysis was. When they discovered electrolysis, they learned how to break compounds into elements, and they discovered a lot more elements that way. So how did it develop over time, as in the periodic table? So this was Mendeleev's periodic table. You can see there's a gap there. That's one of the things Mendeleev managed to do. Now there were some problems that existed in the early periodic table, and this is how Mendeleev solved them. Some elements were in the wrong order because they were arranged by atomic weight. Now our periodic table now is arranged based on proton number, atomic number. So if you look at the periodic table, it increases based on how many protons it has. So carbon has 6 protons, nitrogen has 7 protons, oxygen has 8 protons, so it's in order. Now all they knew at that time was, and Mendeleev's time, is the top number. So carbon would have been 12, nitrogen would have been 14, oxygen 16. So in this case, Mendeleev would have put all 3 in the correct order. But there are other elements where when you put them in order of atomic mass, as in mass number, it doesn't fit a pattern. They end up in the wrong order. So some elements were in the wrong order if arranged by atomic weight. So what Mendeleev did, so by atomic weight, what do we mean? We mean mass number. So Mendeleev changed the order to fit patterns in properties. So if he noticed 2 things had similar reactions, he then made sure they were in the same column, in the same group. Now some elements didn't fit any group, so he left gaps for elements that hadn't been discovered yet. So he noticed that actually there's nothing within all the discovered elements that kind of go into a certain spot, so he left it blank. In fact, what he actually did was he didn't just leave them blank, he predicted the properties of the missing elements, and they were later discovered, and he was very accurate. Now his idea was accepted because elements he predicted were later discovered. So like I said, his predictions were really good. Scientists afterwards learned about isotopes, which proved why he was right to change the order of some elements based on atomic weight. Because it means, for example, you can have chlorine 35, but you could also have chlorine 37. Now if you use chlorine 37, that's going to be in one position on the periodic table. If you use chlorine 35, that would have been in a different position on the periodic table. So afterwards, when scientists learned about isotopes, now what he would have done is he would have put the one that made most sense. So when scientists learned about isotopes, they realized what he was doing was correct. That putting in order of atomic weight, which is the mass number, is not the correct way forward. So today the periodic table is arranged by atomic number. And what's atomic number? That is the number of protons. So now everything is measured in terms of proton number. Right, so let's have a quick look at this exam style question. What is one of the ways in which Mendeleev's periodic table differs from the modern periodic table? So based on everything we've just covered, Mendeleev arranged it by mass number, whereas now we do it based on proton number, the atomic number. Describe how Mendeleev predicted these properties of gallium. So Mendeleev predicted gallium's properties by organizing elements by atomic mass and observing patterns in their properties. So Mendeleev predicted gallium's properties because what he did was he organized elements by atomic mass. So he used mass number, put them in order of mass number, and then he observed patterns in their properties. So for example, he would have put lithium, sodium, and potassium in the same row, in the same column, because he knew they all react in a similar manner. Now, after that, let's say cesium was here, he would have automatically put it here, because he would have noticed it reacts similar to potassium. So the same thing he would have done with gallium. So the table below shows his predictions and actual values. So what he said about, he predicted gallium would be about relative atomic mass of 65. It turned out to be 72. He predicted the density would be 5.5, but actually it was 6.2. So he was close. He said melting point would be lower than 30. It was actually 28.5. And he said the density of its oxide, as in when it becomes gallium oxide, would be 5. But it was actually 5.3. So he was very, very close with all this. Atomic number. Now, atoms of each element have a symbol. There are about 100 different elements in total. Now, what we can learn from this, and I'm going to explain it using this periodic table first before we move on, is that when you go across like this, that is called a period. So hydrogen and helium are period 1. Lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, this is period 2. Sodium, magnesium, aluminium, silicon, phosphorus, sulfur, chlorine, argon are period 3. Now what that teaches us is the number of electron shells it has. So hydrogen is in period 1, it only has 1 shell. Lithium and beryllium are in period 2, so this one here is lithium, this one here is beryllium, they're in period 2 as a second row, so that means they've got 2 electron shells. If you look at the third one, sodium, magnesium, they're in the third period, so they have 1, 2, 3 electron shells. Now, if I was to say rubidium, how many shells would rubidium have? Well, let me just rub some of this, erase some of this, so it's just a little bit clearer. Rubidium is in 1, apologies. Rubidium is in 1, 2, 3, 4, it's in the 5th period. So rubidium would have 1, 2, 3, 4, and 5 electron shells. Now let's look at groups. So that's the ones going down. So this is group 1. This one is group 2. This one here is group 3. This one here is group 4. Group 5. This one's group 6. This one's group 7. And then we've specifically got group 0 here, or group 8. And what that shows us is how many electrons it has in the outer electron shell. So let me get rid of this. And let's understand this properly now. So if I said to you boron is in group 3, what that means is, now boron is in period 2, so it's got 2 electron shells, but it's in group 3. That means it's got 3 electrons in the outer shell. 1, 2, 3. Okay, let's look at chlorine. Chlorine is in period 3. So it's got 3 electron shells. 1, 2, and 3. But it's in group 7. If it's in group 7, it's got how many electrons in the outer shell? 7. So 1, 2, 3, 4, 5, 6, 7. So period 3 means 3 shells. and group 7 means 7 electrons in the outer shell. So the structure of the periodic table. How are elements arranged in the periodic table? They are arranged based on the proton atomic number in rows called periods. Elements with similar properties are grouped together in columns called groups. So if we look here, all of group 1, lithium, sodium, potassium, rubidium, cesium, francium, all have similar properties. Beryllium, magnesium, calcium, strontium, and so on, they all have similar properties. So groups, elements in groups have similar properties. The table is called a periodic table because similar properties repeat at regular intervals. Meaning you go across and then it restarts over here, then it restarts in group 1, it restarts at group 1, and whenever it restarts, it has that same pattern in terms of properties. How does an element's position in the periodic table tell us about its properties? So the period shows the number of electron shells. So this is what we've just explained. The period tells us how many electron shells something has. Non-metals are on the top, are on the right and top. Metals on the left and the bottom. So the split is around here. Oh, sorry, there. Just rub that out. So the split is around here between metals and non-metals. Okay, let's move on. Let's have a go at an exam question. So in figure 1, the letters P, Q, R, S, T and U represent the positions of 6 elements in a periodic table. These letters are not actual symbols for these elements. Now, what I'm going to do really quickly is this. If I was to look at let's look at P. P is in, there's period 1, there's period 2, so it's in period 2. If it's in period 2, how many electron shells does it have? It's got 2 electron shells. It's in group 1, that means it's got 1 electron. So period tells us how many electron shells, but the group tells us how many electrons on the outer shell. So P has 1 electron on the outer shell. So that means P would look like this. Okay, now let's try and answer the questions with all of this information in mind. Write the letters of 2 elements that are non-metals. Okay, so we know that's a non-metal and that's a non-metal. Okay, so what's written here is incorrect. It says write the letters of 2 elements that are non-metals. So it should be T and U. Write the letters of 2 elements in period 2. So this is period 2 here. So you could have written P, S or T. Now Q is not correct because Q is in group 1. So the answer would have been P, S or T. Now, electronic configuration. So the maximum number of electrons you can have in the first shell is 2. So if you look at argon here, how many electrons do you have in the first shell? 2. In the second shell, argon has 8. That's the maximum number of electrons in the second shell. In the third shell, the maximum number of electrons you can add to the third shell is 8 again. So if we look at, let's have a look at sodium as an example. Actually, before we do that, and when we write electronic configuration, how many electrons were in the first shell? 2. How many in the second shell? 8. How many in the third shell? 8 again. So we've got electronic configuration as 288. Now if we look at sodium as an example, it's got 11 protons, so it's got 11 electrons. So it's got 11 electrons. So now if we say there's 2 electrons in the first shell, because that's the max you can put in there, there's now how many electrons left? 9. So what's the maximum we can put in the second shell? It's 8. So let's put 8 there. 1, 2, 3, 4, 5, 6, 7, 8. So we've got 1 electron left. Where do we put that? In the third shell, because we can put 8 there as maximum. So my electronic configuration is 2, 8, 1. So why do hydrogen and helium, what do hydrogen and helium have in common regarding the electron shells? So hydrogen and helium are both in period. So which period would that put them in? Period 1, because they only have 1 shell. And what is the electronic configuration of lithium, beryllium, fluorine, and neon? So the electronic configuration of lithium, how many electrons in the first shell? 2. How many in the second shell? 1. Beryllium, it's 2, 2, because it's 2 in both shells. Fluorine is 2, 7, and neon is 2, 8. Now the fact that we've written down 2 numbers for each one, and there's 2 shells in each one, what period do all of them come into? They're all from period 2. Now, what do all elements in group 1 have in common in the electronic configuration? So if something's in a group, what does the group tell us? It tells us how many electrons they have in the outer shell. So the group number of an element tells you how many electrons are in its outer shell. For example, lithium, it's got 1 electron there, so that means it's group 1. Sodium, it's got 1 electron there, so that means it is group 1. Potassium's got 1 electron in the outer shell, so it is also from group 1. But the difference here is lithium's got 2 electron shells, so that's period 2. Sodium's got 1, 2, 3 shells, so that's period 3. Potassium has 1, 2, 3, and 4 electron shells, so that's period 4. But they are all group 1 because they had 1 outer electron shell, 1 electron in the outer shell. The group number, now what do all elements in group 7 have in common? They've got 7 electrons in the outer shell. So fluorine, his electronic configuration is 2, 7. Chlorine is 2, 8 in the second shell, 8, 7. So what's the difference between the two? They're both in group 7. So that's a similarity, they're both group 7. But from a period perspective, fluorine's got in period 2, it's got 2 electron shells, and chlorine is in period 3, it's got 3 electron shells. Now group 0 is a little bit different. Helium has an electronic configuration of 2. Neon is 2, 8. Argon is 2, 8, 8. And what is similar for all of them is that they all have full outer electron shells, which is why we call it, the best thing to call it is group 0, not group 8. Because it's group 8 from the perspective that neon and argon have got full outer electron shell of 8, but helium does not have 8 electrons outer shell, because in the first shell, the maximum electrons that can go in the first shell is 2. So helium has 2 electrons, but it still has a full outer electron shell. So the main thing about group 0 is they all have a full outer electron shell. So let's have a look at this exam style question. Sodium atoms have 11 electrons. Complete the electronic configuration of the sodium atom. So this is what we did before, so I'm not going to go into too much detail. The electronic configuration of sulfur is 286. Explain how the electronic configuration of sulfur is linked to its period. Now let's do period and group. So what period is it in? It's got 1, 2, 3 numbers, that's period 3. What group is it in? Well, the outer electron shell is 6, so then it's in group 6. So it's in period 3, because it's got 3 electron shells, 2, 8, and 6. Okay, now there's a really good question that's just been asked. What are the predicted topics for 2025? Now that's something that we're going to go through in AMALathon 2. So like I said to you guys before, when we say Amal, so if I say to you, let's Amal this, and I verbalize the word, our company name, what I actually mean by that is to understand everything, to memorize it, and then to apply it. So it's to activate your brain initially, understand everything, and that's what we're doing during this AMALathon. We are activating our brain, we're understanding everything from paper 1 content. Then in the May half-term holidays, we're going to do the exactly same thing. We're going to do the first A from Amal. We're going to activate our brains. We're going to understand everything from the paper 2 content. Now the application side and the part where we'll be going through predicted topics and we'll be looking at past paper questions linked to those predicted topics is what we'll be doing in the pre-exam workshop. So that pre-exam workshop will be the weekend before every single exam, and that's a big part of AMALathon 2. So AMALathon 2 isn't just us going through the content. AMALathon 2 is the weekend before every single exam. We will go through the weekend before every single examination. We will go through all the past paper practice linked to what we think. So Amal has done its own research on this and done its own analysis, what we think is going to be predicted for 2025. So for example, your biology exam is on the 13th of May. We will have a pre-exam AMALathon session on the 10th of May. And that'll be a 3-hour session where we'll cover foundation topics, higher topics, and then we'll have an additional time for triple as well. Okay, now this was where we wanted to get up to. So we are exactly where we want to be at the end of this topic. I want to get up to 1.2. So tomorrow we're going to go through ionic bonding, covalent bonding, and metallic bonding. And then on Wednesday we'll go through all the numeracy stuff. Okay, brilliant. Thank you very much, everyone. We're going to have another 5-minute break, and then we'll have our final physics session. So we'll be back in 5 minutes. We'll restart again at 40. So I'll see you all at 3:40. Thank you, everyone. So you can sign up for AMALathon 2. The link is in the description. Okay, welcome back. Last 1 hour, and we are now on physics. So I hope you can all see me and you can all hear me and you can see the presentation. Got 1 hour to go. Thank you everyone for the 2 hours that you've already given. And we are now going to do our best to get up to 2.15. Now, I'm just going to explain with physics. I have started from P2, and that's because P1 is around 6 slides, and you'll get that for free anyway. And what you'll notice in P1 is essentially it just tells you all the different units for everything. So for that reason, it's not something that I'm going to go through, because I'm going to be teaching you the units as we go through all the physics content anyway. So there's no point me going through a PowerPoint and saying this is the unit for this, this is the unit for this, this is the unit for this. It's just going to be a really boring 10-15 minutes of just reading off the slides. That's something you can do in your own time. So I'm just going to use the time instead to go through P2 in detail. P2 is quite big. It is around, just check, it is around 87 slides. So I'm going to be finishing half today and half of it tomorrow. The aim today is to get to 2.15. This topic has quite a lot of a lot of maths questions, quite a lot of equations. So it's important that you go away and practice those. Now, as you're all aware, you're going to be provided with all the equation on an equation sheet this year, but also keep in mind that you won't be given any units, and that's something that's something to keep in mind. That's something that's really important, something that you remember. You need to know the units for everything, and that's where a lot of students actually lose marks. Okay, so let's get started. So first thing, scalar quantities and vector quantities. You need to know the definition between the two. As in the difference, you need to know the definitions of both and the difference between both. So a scalar quantity has magnitude, which is size, but it does not have direction. For example, distance, speed, time, mass, temperature, energy. Now, vectors, on the other hand, have magnitude and they have direction. So scalar only has magnitude, but a vector has both magnitude and direction. So vector quantities are, for example, displacement. Displacement is a little bit like distance, but look, it's given a direction. It's not just said, I went for a walk and I walked for 5 kilometers. It said I've walked 5 kilometers east. So it tells you direction as well. I moved at 5 meters per second squared. Now the thing with acceleration is that if I'm accelerating, I will say plus 5 meters per second squared. If I'm deaccelerating, then I would say 5 meters per second minus 5 meters per second squared. So we have a direction. Velocity, again, is just like speed, but we've added a direction here. Momentum, we've got a direction. Now, one thing to note here is that velocity is a vector. When you calculate acceleration, it equals the change in velocity divided by time. The fact that velocity is there, or as part of the equation, automatically makes acceleration also a vector. Now momentum, this is momentum, equals mass times velocity. Again, velocity is a vector, so that makes momentum a vector. And force, for example, if someone's falling out of a plane, so that's supposed to be someone skydiving. What is it which way are they falling? Downwards. And it has a size. The thing that's caused them to fall is gravity. It's pulling them down. Now I'm just going to really quickly answer this question. It just said, can I make a predicted topic video? please, it is something that I'm going to be doing, yes. Like I said, if you're able to join us for AMALathon 2, I'll be doing that for 3 hours before each exam. Prior to that, it's unlikely that we'll be able to get a video done in time. So, when we show vector quantities, we show it with arrows. Now, if I show, let me show you 2 different arrows. Here's one arrow. Is that showing the direction a force is acting in? Yes, because the arrow is showing a direction. Now, if I draw a bigger arrow, what's the difference between the first arrow and the second arrow? The second arrow represents something that's bigger. So let's use a car as an example. For the car to go forward, the engine causes an acceleration, so we have a forward force, and I've drawn an arrow this big. So the forward force, it shows the direction of the force, it shows the size of the force because the arrow is quite big. Air resistance, I'm going to show in the opposite direction. So it shows which way the air resistance is occurring. But I've drawn it smaller. So which force is bigger? This one, because the arrow is bigger. So an arrow shows 2 things. It shows us how big the force is, and it also shows us the direction of the force. So differences between scalar and vector. So scalar only has magnitude, which is size, but a vector quantity has magnitude and direction, so it has size and direction. Then we've got our examples. Now at the moment these bits don't matter. But how do we represent a vector quantity and a scalar? Sorry, this bit doesn't matter, but this bit does. Now a scalar quantity, we just write down how big it is. So the mass of someone might be, for example, 50 kilograms. And yes, we'll be covering terminal velocity as well. So that's 50 kilograms. It does come up in the curriculum, terminal velocity. And then when we cover vector quantities, we can add an arrow, and we can also add, for example, 50 newtons. And that shows that it's 50 newtons acting in that direction. So that's magnitude, 50 newtons, and direction, right. So momentum, these are just examples now of different vectors and different scalars. Now this comes up, I've seen this come up in higher papers, and I've seen this come up in foundation papers, and only recently I saw it in a higher paper where all you had to do was say if the quantity was vector or a scalar. So it is important to remember this. So this column, these 2 columns are important. Now these formulas we're going to learn as we go through. We're going to learn these formulas as we go through the different PowerPoints. So velocity. Firstly, what is velocity? Is it a vector or a scalar? And it is a vector. But what about speed? Because sometimes we use the word speed instead of velocity. What is speed? Is it a vector or is it scalar? Speed is a scalar quantity. So velocity is a vector quantity, and it describes the speed of an object. Just going to rub this out so it's not in the way of the writing. Now, unlike speed, so it describes the speed of an object in a specific direction. So it is telling you the speed, but it's also giving you a direction as well. So unlike speed, which only considers how fast something is moving, velocity also includes the direction of movement. So how do we calculate velocity? Velocity is displacement divided by change in time. So you can write that as velocity equals distance divided by time, or you can do velocity equals displacement divided by time. Now, The units for velocity are meters per second. The units for time are seconds. But the units for distance are meters. And the units for displacement is also meters. Usually, we don't, in exam questions in Edexcel, displacement doesn't get used very often. It's usually velocity equals distance over time. So key characteristics of speed. Direction matters. If an object moves 10 meters forward and then moves 10 meters back, its total displacement is 0. So velocity is 0. Now what we mean by this is if someone starts here, if they walk to this friend's house, then they walk to this friend's house and then back to their own house. So let's say friend A, friend B, and then back to where they started. Then what we would say is, and let's say the distance between the houses is 10 meters. So it's 10 meters to that house. And then they come all the way back to their own house. So that's another 10 meters. They then go to their friend B's house, which is 10 meters. And then they come back home. That's 10 meters. So from a distance perspective, how far have they traveled? 10 plus 10 plus 10 plus 10. 40 meters. Now the difference between this and displacement is displacement has a direction. So let me get rid of just this bit here. I will give that man his leg back in just a second. Now, this time when the person walks to friend A, because it's a displacement, it's plus 10 meters. When he returns, it's minus 10 meters because displacement, in displacement we have to indicate the direction. Now from his own home, if he goes this way, he's moved another minus 10 meters because we have to indicate direction as well. And then he returns home. That's now plus 10 meters. So this time, from an S perspective, displacement, the S stands for displacement. What's his overall displacement? 10 minus 10 is 0. Minus 10 is minus 10. Plus this final 10 meters, 0. So we've got 2 plus 10s and 2 minus 10s. So his displacement is 0 meters, meaning from his starting point, he's moved 0 meters. So the difference between distance and displacement is, distance is scalar and displacement is a vector. Let's have a look at this exam style question on velocity. What's the main difference between speed and velocity? Now we're looking at this from a perspective of scalar and vector. Speed is a scalar quantity, so it only has magnitude, and velocity is a vector quantity, so it has magnitude, a size, and it has direction as well. If a car moves 100 meters east in 5 seconds, what's its velocity? So how do we work this out? We work out velocity by doing distance divided by time. So our distance in this case is 100 and we're dividing it by time, which is 5. So 100 divided by 5. Now here we've been given east as well, so we can add that as part of our answer. So the answer equals 100 divided by 5 is 20, so it's 20 meters per second east. But can an object have constant speed but changing velocity? Yes, it can. Now let me explain why. This is something that will come up afterwards as well. But let's say you've got, actually let's look at the planet Earth. There's Earth and there's the moon. Now, the moon is going around in a circular position. Let's say the moon's going at, it's much faster than this, but I'm just using an example, 100 meters per second. That's its speed. Now, what is it constantly doing? What's it constantly doing at the same time? It's constantly changing direction. So it's there, then it's there, then it's there. Now, is it constant speed? Now, if we're talking about constant speed, it's got constant speed. Let's say it's just only going at 100 meters per second, but constantly it's changing direction. So, because it's constantly changing direction, it has to be velocity. Because now we're talking about size and we're talking about direction. So that means because it's changing direction constantly, it's also changing velocity, even though the speed stays the same. So yes, if an object moves in a circular path, its speed remains the same, but direction changes, meaning velocity changes. Now I just want to clarify one thing here. Velocity is generally measured in meters per second. However, that part of the unit there, meters, has to correspond to that unit there. So for example, if you're told the distance is in kilometers, then you can measure the velocity also in kilometers per second. And the second part where it says seconds, that has to correspond to the time taken. So if it says, for example, kilometers per hour, and it says per hour, then you can treat the time as hour as well. So they have to correspond to each other. But in general, the questions that you're going to get are meters per second, meters, and seconds. Okay, let's continue. So, okay, what is speed and how is it calculated? So I'm going to give you a second just to read this. So speed is a scalar quantity that refers to how fast an object moves, regardless of direction. It only measures the magnitude of motion and does not consider direction. So speed equals distance over time. So this is the main formula that we're going to be working with. Speed equals distance over time. Because these are all scalar quantities. When we talk about velocity, velocity equals displacement divided by time. Speed direction is not considered. If a car moves 50 kilometers east or west, its speed remains the same because it's a scalar quantity. If it says 50 kilometers east, then we're saying that we're talking about a vector quantity. So what is the main difference between speed and velocity? Speed is a scalar quantity, while velocity is a vector quantity. So it has magnitude and it has direction as well. Okay, so let's have a quick go at this question. It is nice and it is a nice simple question. If a train covers 200 kilometers in 4 hours. So this is what I was saying, you've got to keep the units consistent. You don't have to do conversions. In a lot of science, you have to convert the units first, but in speed, you don't always have to. So we've already said speed is distance divided by time. So that means in this case, we're saying 200 divided by 4. Which is 50. And because we've got kilometers as our distance and time in hours, we can put our units afterwards as kilometers per hour. Can an object have high speed but zero velocity? So that's the question you don't need to worry about too much. Now, this is something that comes up in many exams. It's important to understand instantaneous speed versus average speed. So instantaneous speed is the speed of an object at that specific moment in time. It's what a speedometer in a car shows at any given instant. I have seen 2-mark questions on this, which ask you to compare the two. So when you're in your car, and now you might have 2 different types of cars. You might have one car that just says 26, for example, and that means how fast it's going. That's instantaneous speed. At that one moment in time, it's going at 26 miles per hour. Or you might have a car that's got numbers like this, and it has an arrow, and it's pointing to 26. So you know it's going at 26 miles per hour. That's instantaneous speed. Whereas average speed, now you don't need to know this part here. Now with average speed is the total distance traveled divided by the total time taken. It gives an overall measure of how fast an object moved over a period of time. So this is where we would say average speed equals total distance divided by total time. So if a person, for example, so here as an example, a cyclist travels 60 kilometers in 2 hours. So we're going to do 60 divided by 2, 30 kilometers per hour. But another example could be a person travels at 60 kilometers for 1 hour, but then they have a rest for 1 hour. Then they travel at, now, actually no, we won't bring this up yet. We'll come back to this kind of question. Okay, now, numerical questions for instantaneous and average speed. So a car covers 5 meters in 0.2 seconds. What is its instantaneous speed at that moment? So remember, speed equals distance divided by time. So we're doing 5 divided by 0.2, and that equals 25 meters per second. But that's how fast that car is going at that moment in time. Now a ball is dropped from a height, and its speed after 3 seconds is 29.4 meters per second. What is the ball's instantaneous speed at that moment? Now this particular question I'm going to leave there, but I will come back to it when we cover it, because V equals U plus AT. So when something falls, for example a ball is dropped, I'll explain some of the science behind this. That ball, when it's falling at an acceleration of 9.8 meters per second, meaning gravity is causing it to accelerate by 9.8 meters per second. And so that means A equals 9.8. Time is 3 seconds, and V is the final speed. So after 3 seconds, it's recorded as 29.8. So it's 29.4. And U is its initial speed. So if you were to calculate all this, you would end up with, a ball is dropped from height, so its initial speed would be 0. Plus, and then for A, you've got 9.8, and time you've got 3. If you multiply them together, you get 29.4. And that shows that the final velocity is 29.4. But we will cover this particular formula in a lot more detail. So everything we're covering right now is both in the foundation exam and in the higher exam. If anything comes up that's specifically higher or specifically triple, then I will let you know. Okay, I'm going to move past these exam style questions. Once you download the notes, you will have access to them, so you can have a go in your own time as well. And what I would advise is that you start looking through past paper questions. So if you're spending 3 hours with myself, looking through one exam paper per day or spending a little bit of time on exam practice would be beneficial, but I understand you've got lots of other revision to do as well. So a distance time graph is a visual representation of how the distance traveled by an object changes with time. It helps in understanding the motion of an object, including whether it's at rest, moving at a constant speed, or accelerating. So this is what you need to look at. So you've got a distance time graph here, and we need to understand every part of this distance time graph. So the x-axis represents time, that's shown here. The y-axis represents distance. Now, if you have a line that is at a certain angle, that represents constant velocity. If you've got a flat line, that means because the distance is no longer changing and the person's distance has stopped, that means the person has also stopped. So horizontal line indicates the object is at rest, it's not moving anymore. So an angled line represents speed, represents constant speed, so uniform motion. If you have a horizontal line that is at a steeper angle, that means the person is still moving at constant speed, but it's a greater speed than the other line. So the greater the gradient, the faster the object is traveling. The horizontal line means it's going at one speed. And a curved line, which is not on this particular diagram, and it's very rare, I've not actually seen it in an exam. Or I think I've seen it once in an exam. The curved line represents acceleration, but that's very common. And a curved line going downwards represents deceleration. So let's have a look at an exam style question. So how can these scenarios be represented on a distance time graph? So what does this straight line represent? It represents a stationary object. What does this line represent? It represents constant speed. Now, both of these lines represent constant speed, but the steeper gradient represents constant faster speed. And if the line is going downwards, that represents constant speed backwards. It's when a person is returning home. And the curved line represents acceleration, and a curved line going upwards. Because what you can see here is that the speed is slowing down, so that represents deceleration. If the line does this, it means the person is returning home, but the speed's getting slower and slower. But that is unlikely to come in exam, so that is something that I'll rub out because it is irrelevant to your learning. So what you get here, between A and B, what's happening? The person is traveling at constant speed. But from B to C, the person is still traveling at constant speed, but they're traveling at a faster speed because the gradient is steeper. And from C to D, they've now returned back to that lower constant speed. Okay, calculating acceleration. Now there's a question here which I'm just going to really quickly answer, and it is for biology is vasodilation in Edexcel. I know this is a physics topic, but I'll quickly answer this. It is for biology triple award. Vasodilation does come into homeostasis, but that's something that will get covered in our next AMALathon. So just to recap, our next AMALathon, so AMALathon part 2, will start in May, where we'll have a week of learning just like this, where we'll cover all of topic 2. So we'll be activating the brain, understanding everything in the 1 week holidays, in the May half-term holidays, and we'll cover all of paper 2 content. Alongside that, we'll be doing pre-exam past paper topics. So like a pre-exam workshop where we'll be going through like a predicted paper almost for 3 hours prior to the exam. So if the exams, for example, biology is on the 30th of May, on the 10th of May, I'll be doing that pre-exam workshop. So if you want to register for that, the link is below. Okay, now calculating acceleration. What is acceleration? So acceleration is the rate at which the velocity of an object changes over time. It's a vector quantity. So what we're saying here is, let's say someone's speed so let's say we've got a person's got a speed of 2 meters per second, then it increases to 4 meters per second, then it increases to 6 meters per second, then it increases to 8 meters per second and 10 meters per second. That means it has an acceleration of 2 meters per second squared, meaning every second the speed is increasing by 2, and that's what acceleration is. So let me recap that. If someone's speed is 2 meters per second, then increases to 4, 6, 8, 10, the speed's increased by 2 each time. That means the car has a uniform acceleration of 2 meters per second squared, because that is the units for acceleration. Now, acceleration is calculated by finding out the change in velocity divided by time. So one way of doing that is you find out the final velocity, which is v, and from that you take away the initial velocity, which is u. So v minus u, and then you divide that by the time. Okay, you don't need to know about zero acceleration. But so here's the question. Well, just to give everyone a chance to apply that learning. So we've got a car that accelerates at 10 meters per second to 30 meters per second in 5 seconds. So that means, so remember, A equals V minus U divided by T. So in this scenario, what's V? It's 30. From that we take away U, which is the initial speed, 10, divided by the time, which is 5. So 30 take away 10 is 20, divided by 5 we've got 4. And what's the units for acceleration? Meters per second squared. So the car's acceleration is 4 meters per second squared. A bike moving at 15 meters per second comes to rest in 3 seconds. Find its acceleration. Now, in this situation, in this question, what's the initial speed? If something is at rest, then I'm just going to do the working out for 2 here. What does V equal? That's a bike's moving at 15 meters per second, so it's 15. What's U? U is its initial speed. Now it started from rest. At rest, our speed is 0. What's the time? 3. So we're doing, no, sorry, I've said this the wrong way around. Initially it's at rest. Oops. Right, my fault. So initially, a bike is moving at 15 meters per second. So our initial speed is 15. Then it slows down until it comes to rest. So the final speed is 0 and our time is 3. That's why we've got 0, our initial speed, so our final speed, minus our minus 15, which is our initial speed, which is minus 15, divided by 3. So that means that the answer is, acceleration is minus 5 meters per second squared. In other words, the bike has deaccelerated. And that's why it's a vector, because you can have a positive answer or a negative answer. And then the final question, a cyclist moving at 10 meters per second slows down to 4 meters per second. So what's the cyclist's initial speed? So let's look at number 3 over here. Initial speed is u, which is 10. Our final speed is 4, and it takes 3 seconds. So time is 3 seconds, which is why we're doing 4 take away 10 divided by 3. So 4 take away 10 is minus 6 divided by 3, and we have got an answer of minus 2 meters per second. So the cyclist has a deacceleration of minus 2 meters per second squared. Calculating acceleration. So what is uniform acceleration? Uniform acceleration occurs when an object's velocity changes at constant rate per second. So this is what I was showing you before. If the speed goes from 2 meters per second, then it increases to 4 meters per second, then 6, then 8, then 10. The acceleration is 2, because it's increasing by 2 meters per second. So that's uniform acceleration, because the speed's increasing by the same amount each time. And that would be the same if it was slowing down. Now, another formula to work acceleration is v equals u plus at, acceleration times time. So here we know that v is our final speed. Now this kind of question usually comes in the higher paper. It's unlikely to come in a foundation paper. But v is our initial speed, u is our final speed. So u is our initial speed, v is our final speed, plus acceleration times time. So let's have a look at an exam question. A bike is moving, so final speed equals initial speed plus acceleration times time. So here we've got a bike moving at 10 meters per second. It accelerates at 4 meters per second. What will its speed be after 5 seconds? So this is u, this is the initial speed. This is the acceleration. And here we have got time. Now because this question doesn't require rearranging, it's more likely to be a foundation question. So u equals 10. And to that we're adding a and t multiplied by each other. So 4 times 5. So that equals 20. So 10 plus 20, 30 meters per second. Now, velocity time graphs. A velocity time graph represents how an object's velocity changes over time. The x-axis shows time, and the y-axis shows velocity meters per second. These graphs help analyze an object's motion, including acceleration and distance traveled. Now this is probably best explained through these. So that line that's flat at the bottom there, that means it has a velocity of 0. If something has a velocity of 0, so with the velocity time graph, we've got velocity on the y-axis and we've got time on the x-axis. Now just so you're aware, distance time graphs are more likely to come up on a foundation paper. But a velocity time graph is more likely to come on the higher paper. Having said that, last year a distance time graph came up in the 2024 paper, and it came in the higher paper. So it does not mean that it won't come up, and it doesn't mean that velocity time graph can't come in the foundation paper. It's just more likely. That was the first time a distance time graph came up in the foundation, in the higher paper as well. So it can happen, and it was the first question, meaning it was one of the last questions. The distance time graph came up as one of the last questions in the foundation paper, but it came as one of the first questions in the higher paper. So it can come in both. Now, a velocity time graph, this line at the bottom shows that we have as a velocity of 0. So that's a stationary object. Now, here it shows that the velocity is staying the same. So that would be constant velocity. If the line's going downwards, that means the speed is decreasing. And if you look at it, it's a straight line, that means it's decreasing at a constant rate. So that is constant deceleration. And if you want to show constant acceleration, it would be a line going upwards. And here again, you can see that's uniform constant acceleration because it's increasing by the same amount per second. It's a straight line. Now, the way to calculate acceleration from a velocity time graph is by calculating its gradient. So that means the change in y divided by the change in x. So this is purely a maths question. And don't forget, in a physics exam, you're looking up to 40%, sorry, up to around 30% of the exam. So in combined, that means around 18 marks or 24 marks is all maths. I've seen more in some papers and less in some papers. So it's always between 18 and around 18 to 24. So if you're doing triple award, you're talking 30 plus marks will just be maths. So you already get yourself into the grade 4 category just from doing all the maths questions. So that's a good way to revise. Now, if we're trying to work out the acceleration of A to B, then our change in y, in other words, so that just treat that as a triangle. From here to here, our change in y equals 18. I'm going to divide that by the change in x, so that is 6. So what's the acceleration from A to B? It is 3 meters per second. Now, this is one of the core practicals, or is a practical based topic. Now, like I said to you guys before, I can't cover core practicals in more detail beyond the theory. So what I would advise is whenever a core practical comes up, YouTube, do a YouTube search and watch one of the animations, or watch one of the YouTube clips that are linked to that particular practical. So usually if you go to for example, the Edexcel YouTube channel, I think it's been made by Pearson's, because Edexcel is owned by Pearson's, then you'll find lots of practicals that have been made specifically for the Edexcel exam board. So here we've got a car. And that car is on a ramp. So we've got a toy car, a ramp. We've already used a meter ruler to see what the distance from one part to the next is. So we know from here to here is a meter. We've got stopwatch to time from when it starts moving till it goes past a specific point. Now, better than using a timer would be to add light gates. So if we have light gates, as soon as it cuts across the light gates from either side, it automatically times for you, and that's more accurate than a stopwatch. The issue with the stopwatch is, I'm sure a lot of you know this, it is our reaction speed. So when we react to things, it sometimes takes up to 0.3 seconds, and that's extra time that we're adding on. So you can't end up determining the incorrect speed because of how slowly we react to things. So you release the toy car, let the car move from A to B without pushing. So you let gravity cause the acceleration. Now here are the steps. We set up the ramp, tilt one end so the car rolls down due to gravity. Mark A to B, so use a ruler to mark the start point as A and the end point of B. So that way with the ruler you also know what the distance is. It could be half a meter, 2 meters, whatever you've decided. Then you get the stopwatch ready. Prepare to time the car's motion. As second it goes past A, start the stopwatch. When it goes past B, stop the stopwatch. Or if you've got light gates, it does it automatically for you. Release the car, in other words, don't touch it, as in let go, without pushing it, let it roll freely from A to B. Time the motion. Do several trials and average the times. So when you do something the first time, the second time, the third time, the fourth time, if you've got, for example, 10 seconds, 19 seconds, 9 seconds, and 10 seconds, then you know there was an issue here. So you delete the anomalous result. Then you get your average. Then you do 10 plus 9 plus 10 and then divide that by 3. So you ignore the second one. So whenever you have data which doesn't fit, you delete it. It's an anomalous result. So before you work out averages, you've got to delete your anomalous results. And then you can calculate speed and acceleration. So remember, it's distance divided by time. And we know the distance and we know the time it just took. Now you can do final velocity minus initial velocity divided by time as well to work out the acceleration. So the kind of questions you sometimes get alongside practicals are identifying and minimizing risks in an experiment. So what potential risks are involved in this experiment? How can they be minimized? So you've got a fast moving toy car. The car may fall off the ramp and cause damage. So you can use barriers or edges to prevent falls. On unstable ramp, the ramp might collapse during the experiment, so you need to secure the ramp firmly before starting. Inaccurate timing. Inaccurate timing with the stopwatch. Inaccurate measurements can affect the results. So you take multiple readings and calculate an average. The moving car could hit someone, so the car may roll off and hit an observer. So you would conduct the experiment in a clear space. What alternative equipment can be used to measure the time more accurately? Like we've already mentioned, you would use a light gate or an electronic timer instead of the stopwatch, and that would get rid of our reaction time. That would make it a lot more accurate. Now, light gates are infrared sensors and they detect when the toy car passes through automatically. So that means you'll have a light sensor here and a light sensor here. So it knows exactly when it's gone through, and that then automatically works out the speed for you, automatically works out the acceleration for you as well. Now the reason they improve things is what's written here. For myself, I would always advise to write about light gates. Electronic timer generally doesn't come up as part of a mark scheme, although it will still be a correct answer. Now these alternatives improve precision by eliminating reaction time delays. So it gets rid of reaction time. That's the main thing here. So ensuring the validity, so that should say validity of experimental results. How can we ensure our results are valid? So number one, repeat the experiment. Do multiple trials. That gets rid of outliers or anomalous results, and it ensures the results are reliable. Once you spot an outlier or anomalous result, you delete it. You can use accurate tools, so use light gates because they're more precise than stopwatches. Stick to the same method, follow the same steps each time. Control variables for this experiment would be keep the ramp height the same, keep the surface the same. Because if you change the surface, you can have different levels of friction. If you increase the height, then by increasing the height, so if it's that height and you increase it that height, you now have gravity acting at a different angle, so it will increase the acceleration. You need to use the same toy car throughout, because again, if you use a different toy car, you've got different frictions, they might have different tires, and you start from the exact same position each time. So if you look at where they were starting from, you start at that point every single time. Okay, typical speeds. So this actually answers a question that's just been asked. Do we need to memorize the typical speeds, such as walking and running? Now, it does come up as a question, particularly in foundation. So I was looking at the foundation 2024 paper, and there was a question linked to this. It was a multiple choice question, however. What I would advise is, typically a walking person's speed is 1.5, a running meters per second. Running is 5 meters per second. Cycling is faster. Car in city is 13. Now, you don't need to know in this much detail. They are usually more common sense type questions. So if it says, what out of the four answers, what is most likely to be the walking speed of a person, and it says 0.05 meters per second, 3 meters per second, 20 meters per second, and 200 meters per second. And this is the way the question was written in the question. Now, that's too slow, traveling 0.05 meters per second. 20 meters per second is really long. That's the length of people's houses. So no one travels that fast in a second, and 200 meters per second, no one travels that fast in a second. So it can only be 3 meters per second. So what I would advise is you have to approach these kind of questions with common sense, rather than wasting time memorizing all these numbers. And then additionally, all these kind of speeds would change based on a person's age, fitness level, etc. Free fall acceleration. So this is linked to terminal velocity. So free fall now, I am going to finish up to 2.15. So if you've all got time, I am going to continue for another 5 or 6 minutes. If anyone obviously needs to log off, I'm not. It is live and you can watch it afterwards anyway. Now, free fall is the acceleration of an object due to gravity alone, so no air resistance. Meaning if you drop a ball, what's making it accelerate? It is gravity, which equals 9.8 meters per second squared. Meaning every second, it increases the velocity by 9.8. So if we use the example of 10, let's round up to 10. That means if someone jumps out of a plane, after 1 second, their speed will be 10 meters per second. After 2 seconds, their speed will be 20 meters per second. So what will their speed be after 5 seconds? It'll be 50 meters per second. So, key concepts of free fall. Objects that are in free fall accelerate at 9.8 meters per second squared. Mass doesn't matter, because objects fall at the same rate as long as there's no air resistance. Because if you've got more air resistance, that means you've got more air particles. Every time this ball, for example, hits an air particle, the air particle exerts a tiny force in the opposite direction. So the more air resistance, the more air there is that a person falls through. In other words, the faster a person's moving, the more air they're going to fall through. then the greater the air resistance becomes. Because if you're falling through, you're colliding with more air, you're going to have more air resistance hitting you. This, we don't need to know, that should not be there. Now, when someone starts to fall, I'm just going to show you on this slide here, and then I'll show you on this slide. So when an object initially starts to fall, it has acceleration, so its speed starts to increase. Now, the faster the person gets, what starts to happen is the person's speed increases, they're falling through more air, so they have more air particles causing a force in the opposite direction. So now the air resistance causes a force in the opposite direction until the person travels so fast that the air resistance becomes the same size as the acceleration pulling it downwards. Now at this point, when gravity, or we could call this weight, is the same as air resistance, at this point the person can no longer accelerate, because we can only accelerate if one force is bigger than the other. At this point, we now have reached terminal velocity, maximum velocity. We've reached the point that the person can no longer accelerate faster than, so the speed will not increase beyond this point. So we've reached maximum speed, and it's a constant speed. That's because there's no force. You need one force to be greater to cause acceleration. Okay, 2.14, Newton's first law. So an object stays at rest or moves at constant speed in a straight line unless acted on by a force. So what we mean by this is, if this is an object, if I want to cause this object to accelerate, I've got to put a force going in this direction, and now it will accelerate towards the right. Now, if the force in the opposite direction is the same size, then can that object accelerate? It can't accelerate, because now neither force is bigger. So now there's two things that can be happening at this point. Either that object is now moving at constant velocity, so it has a constant velocity, because whatever speed it was at, it will just stay at that speed, because if this force here is not bigger, it's not going to accelerate. If this force here is not bigger, it's not going to de-accelerate. So velocity just stays the same, constant velocity. And there's one more scenario. If you have an object and the forces are equal, then it can also mean that the object is stationary, because all the forces are equal. So let's quickly read through what's written here. So if there's no unbalanced force, motion does not change, meaning one force has to be bigger than the other. Now, objects keep moving without stopping unless something acts on them. So in cars, seat belts and airbags protect us by dealing with inertia in sudden stops. What that means is, if we're moving this way and we collide into something, then it exerts a force in the opposite direction, which is the same size. And so if we're in a car and we keep moving, but there's a seatbelt attached to a person, when that car suddenly stops for whatever reason, let's say it hits something, that person is still going to continue moving at that speed. So what the seatbelt does, it exerts a force in the opposite direction to stop us from going through, so it causes us to become stationary. So what will happen to the motion of each of these cars and why? The motion of a car depends on the forces acting on it. So if there's no external force acting on a moving car, it will continue moving at the same speed in the same direction. So this is what I was explaining before. If you've got a force going this way, it'll accelerate. If the force in the opposite direction is the same size, then there's nothing to cause it to accelerate from this side, and there's nothing from the left hand side to cause it to slow down. So the car will just go at the same speed. Or it can also mean that the car is stationary. So if an external force like brakes or friction act on it, its motion will change. It may slow down, stop, or change direction. So now, if we increase the size of the arrow here, then we'll cause the car to start slowing down, or the object to start slowing down. Okay, so from tomorrow, so firstly, thank you everyone for being in the session for the last 3 hours. It's been a really good session. Hope you've all enjoyed it. If you're able to give me some feedback on what you might want to do differently tomorrow, or if you want to give me a rating out of 5, I really appreciate that. Now there was a question here. It says, is there a course for GCSE maths? Unfortunately, there won't be one this year, but as AMAL education increases and grows, we will start doing maths and English, particularly from September. Well, if anyone's here from year 10, we'll be doing something called the weekly AMAL. What that means is we'll have English, maths and science tuition on YouTube live on a weekly basis. So if you make sure if you've joined our WhatsApp group or WhatsApp channel, or if you've registered or you subscribe to our YouTube channel, then that's probably the best way to keep updated with everything. Okay guys, have a lovely evening. I'll see you all tomorrow.